Good morning, sir. Aishwari, ma'am.
Hello.
Sir, good morning.
Good morning, madam.
How are you, sir?
Fine, fine.
The Dean ma'am join Marathon, sir. Then we can start. She'll be joining now, sir.
Good morning, one and all present here. Big journey begins with small steps. We, Swarnalakshmi and Rajarajeshwari, research scholar, Department of Botany, Avinashilingam Institute for Home Science and Higher Education for Women, Coimbatore, are privileged in mastering the ceremony of UGC stride.
Raji? Raji, Dean ma'am join Marathon.
One song can change a moment. One idea can change a world. One step can start a journey. But a prayer can change everything impossible.
Raji?
Hello, ma'am.
Swarna? Dean ma'am join Marathon. You wait for 2 minutes.
Okay, ma'am. Yes, ma'am. Sure, ma'am.
Raji? Ma'am join Marathon. Sir,
Good
Dean
morning,
ma'am
one
has
and all
joined the
present.
link, sir. Okay, you can start.
Good morning, one and all present here. Big journey begins with small steps. We are Swarnalakshmi and Rajarajeshwari, research scholar, Department of Botany, Avinashilingam Institute for Home Science and Higher Education for Women, Coimbatore, are privileged in mastering the ceremony of UGC stride.
One song can change a moment. One idea can change a world. One step can start a journey. But a prayer can change even the impossible. With this, we'll begin this program with a prayer song. Now, I would like to invite Yangchen Dolmakom, research scholar, Department of Botany, Avinashilingam Institute for Home Science and Higher Education for Women, Coimbatore, to sing the prayer song.
भूमनाति जानता देवा वा गम्यता पूर्वे समानानां वासते समान हृदयानिहं समानमस्तु मनु यथा वस्तु सहसते.
Thank you, Yangchen, for your soulful prayer song.
Welcome to those who believe in the power of dreams and who would like to join me in the exploration of life. I welcome all the dignitaries, deans, HODs, faculty members, research scholars, and students of various departments to this virtual program.
Now, we will begin the technical session by introducing the resource person, Dr. Debaprada Das, Assistant Professor, Scientist 3 and Head Department of Botany, Shiv Bharati Mahavidyalaya, West Bengal. He acted as a government officer since 2002 under West Bengal Education Service. He was a research fellow of Forest Productivity Project funded by ICFRE, Dehradun, and done his work at Southwest Bengal Forest from Central for Natural Studies at Raja L.N. Khan Women's College, Madhyapur, West Bengal. Later on, he worked as JRF and SRF at Central National Herbarium, Botanical Survey of India, Singapore, West Bengal, under Flora India project. Now he is a coordinator of a service lecture and conference of many national and international kinds. He also authored more than 100 scientific research papers and 4 books in environmental studies and in botany, including biodiversity and sustainable management. Now he is here to share his expertise on the topic ecological assessment of healthy environment throughout vegetation study. We are fortunate for having you here, sir. The session is over to you, sir.
Thank you. Thank you, madam. Good morning, one and all. I am thankful to the organizers for giving me this opportunity. This is a 6-day long training program on environmental sustainability and research ethics, sponsored by UGC STRIDE. Very important wings of the UGC that declare this one just a few months before the COVID period, and organized by School of Biosciences, Avinashilingam Institute for Home Science and Higher Education for Women, deemed to be university. I am thankful all the organizing team members, Chancellor Sir, Vice Chancellor Ma'am, Dean School of Biosciences, and head and faculty of different departments, even research scholars and students. So today I am giving to present one that is talk entitled Ecological Assessment of Healthy Environment through Vegetation Study. So this is related to one that is domain research, and that always associated with various subdisciplines. It may be biological, it may be physical, it may be social, it may be anthropological, and so on. You can apply this technique everywhere to know your environment that is better. Quality-based nature, quality-based environment, quality-based ecosystem, quality-based tuned that is surroundings that we need, because we need the green environment, green university campus, clean university campus, free from plastic, polythenes, non-biodegradable products, and we have to manage. So today's presentation is ecological assessment. You know, different terms applied here: nature, environment, ecosystem, ecology, vegetation, assessment, and ultimately assessments of healthy ecosystem. And if you go to this study, then obviously you should have to know the science, because each and every event having the science background, and behind it the explanation obviously by the researcher or the study persons from the field. It may be the layman, it may be the scholar, scientist, teachers. Everyone can assess, and this is based on the explanations and ultimately applications. So your explanation, your research, your observations should have to place in a particular place to enhance the quality. This is called application and reassessment, and after that validations. So every time we need the validations. So without wasting time, I am going to the presentation. So madam, can I share my slides? Madam, can I share my PPT slides?
Yes, sir. Please
Okay.
share it, sir.
Okay.
Yes, sir.
So I think now in slides are visible.
Yes, sir.
This is network issue. So okay, now fine. So now fine. My topic is ecological assessment of healthy environment through vegetation study. So this is a slide that you see. It is the picture taken from the hill of our Darjeeling, and it is Sonada nursery. And this is a nursery that is governed by Government of West Bengal, Department of Forest. This nursery having various types of plants, but this particular nursery net houses for Michaelia, a very important species of the hill. And you see the forest worker, the forest departmental that is official that is here and nurturing plants. That this series, this left hand side of the series of the plants are juvenile one to higher order of plants, and having the mixtures of the growth and even the different parameters like diameter, height, and etc. And right hand side, the plants are heavier growth. So every time that need monitoring. So we just pick up the plants of the healthy one from the unhealthy part of the site to the healthy sites, and always measuring the height, that is diameter, breast, any infection or not. Then just isolating and collecting the stock for better growth, better healthy stock, for better environment and better sustainability, ultimately for better that is output. And after that, obviously within 2 to 3 years, it will be go to the field for placement in university campus, in college campus, in institutional campus, in roadside, in some hilly areas where that is required. But my particular point is that if you just go through the study of nature, environment, ecosystem, ecology, vegetation, assessment, and assessments of healthy ecosystem, obviously you need the science, explanation, application, and assessment. Every research, every science that having the science, that having the background of the science and explanation, you explain that is non-healthy, that is healthy, and this is unhealthy due to the infection by various disease-causing organisms or due to the physical I think parameters. It may be high temperature, high snow, high rainfall, high that is wind blow, or it may be infection by various types of bacteria, fungi, nematodes, molds, and so on. So this is this is a complexity, and this is a natural means to always make abnormality regarding the degradations of the normality. So always change that is abnormality from the normality. So our train to retrain from abnormality to normality to regain back to the normality. So they are monitoring and they are assessing and everywhere. So let's go for other slides. So this is nature. You know, nature is unique. Nature means the universe. This is the physical world collectively, including plants, animals, landscape, and other features like inanimate bodies, or it may be animate and inanimate bodies other than the creative features. It's called a nature. So this is universe and created by our God, or it may be Allah, or it may be by Bhagavan, whatever it may be, the uniqueness, the embodied human creations, nature, and it refers to the phenomena of the physical world. An environment, that is our surroundings or conditions in which a person or animal or microorganisms or plants or any other organisms thrives there and operates their activities. You see this figure that having the second one, that having the bird, that this is kingfisher, and having the poppy plant. So this is the scenario. So if poppies are not here and again filled with the sunflower, then automatically parakeets and parrots will come here. So the sitting arrangement is same, but no kingfisher, but other birds like parakeets and parrots rather than the kingfishers, and no poppies are here regarding the placements of sunflower instead of the poppies. So this is the environment. So environment is changing every time, and that is why we are called that is non-linear, and non-linear dynamics is always going on in nature. That's why we have to study day by day, month wise, or year wise, or repeatedly year after another years. So these are ecosystems, and simply that is geographical area where plants, animals, other microorganisms are working together, as well as weather, landscape, and together forms a bubbles of life. And if you consider this is a particular land habit, that ecosystems is a specific kind. If it is a aquatic body, it is a habitat for other organisms along with the terrestrial bodies. So habitat and structure, patterns, ambiguity, all are different time to time due to change of the physical parameters. An ecosystem contain biotic or non-living part, biotic and living part, and non-living part or abiotic part as a whole for factorial change. And this is a picture that having the coastal part, having mangroves and others with the aquatic bodies. So various kinds of organisms are there. If it is in sweet water, then automatically sweet water animals or organisms will be there. If it is brackish, or if it is marine, so marine kinds of organisms will be there. So different. So in a study, we can say that ecosystem study is called ecology. So ecology is the study of the organisms and how they interact with the environment around them. So this figure having the energy, different parameters like live with chlorophyll, different energy cycles, and so on, all in an integral way. And this one having the units, it may be lake, it may be a forest, it may be trophic structures, it may be food, it may be any kinds of energy level transfer conditions, and so on. So in any ecosystems, ecological parameters are different, but the basic themes are same. So this is first book written by German that is scientist Ritter in 1885, ecology, and from that plant scientist and environmentalist on plant physiology parameter based book published first time in ecology, that is plant ecology, 1895. So these are the two pioneers in this field. So you see, this is a picture that having the that is desert plants, having the desert that is habitats, and this having also the desert, but it may be somehow different from the desert. So these two uniqueness are here, this figure and this figure. And this is alluvial plain, having no water. So plants are, that is planted kind, that is maize plant. So maize are here along with no rainwater, no water. So this is drought. So need the drought resistance, you see. So vegetation is drought tolerant C4 plant, you know C4 plant. So these are different, this is different. But this one is that is a lower part of the waterlogged condition, that is farm vegetation, great farm dominated vegetation, and this is mixed vegetation, and this is green or evergreen vegetation. So everywhere that is vegetation types are different, that depends upon the habitat. Now assessment, we need the assessment. Why assessment? Assessment because we need to know the actions which is going on. The actions or assessing something is called assessment. The method that is employed on the assessments is methodological placements. You can examine, you can interview, you can take interview, or you can do the test. The examine methods is process, reviewing, inspecting, observing, studying or analyzing the material. Assessment objects are specific object, but mechanisms are same. So activities regarding this type of assessment is nature, condition strategy or strategy study or situations over the time. This is a good book. You can take this reference for your particular study. Jorgensen and others authors edited this book. This is ecological indicators for the assessments of ecosystem, health ecosystem. So other related that is it may be literatures or books are here. You can consult. But you come here, assessment, environment or ecosystem assessment. Particularly two types of assessments are here. That is national level ecosystem assessments and regional level ecosystem assessment. Regional level assessments are based on the centers at Delhi or Pune or Mumbai or Mangaluru, sometimes Chennai, Kolkata, Ahmedabad or Hyderabad, that is nearby your institution. You just make some unit. Here 4 units are there. That is technical unit, review unit, assessment unit, verification unit, but all need the validation. So obviously we need one unit, that is validation unit. So you can assess through validation. So validation unit having the technical units and review units, and every time that need assessment. So assessment unit and verification unit as a whole can call the validation unit. So we can establish validation unit in our institute, in our college, in our university, or in our NGO that can help us to monitor the environment time to time. You know, this is health and health of ecosystem. This figure having the good lilies and nymphoids and nymphaeas, but here this situation is breakdowns into heavy depositions of plastics and similar materials, and plastic containers having the chemicals and pesticides. So automatically chemicals and pesticides are releasing to the water, and the water quality is gradually deteriorating, and that lead to the death of the organisms and even the death of the plant organisms above the surface of the water or underneath of the water. So automatically this will go to that situation that having no green vegetations. This duck is here, but having no materials to collect her food. So this is a complex situation. So you can easily say this environment is healthy environment. This environment is deteriorating, and this is a bad environment. So in this way, you can assess superficie, and if this environment is good, that is bad, that is too bad, and so on. But in a scientific way, we need the degree, or that is maybe data for assessing the vegetation, for assessing the environment, or assessing the ecosystem. That is tuned one. This figure is a degraded versus natural forest. This is cutting the vegetations under the slopes. This is a green patch having no loss of the vegetation, having lichens, mosses, and others. So that is natural one. This one is ruined one and gradually diminishing because no ground vegetation is here, and continuous ground flowers emitting the greenhouse gases, particularly carbon dioxide, carbon monoxides, and some places sulfur oxides and nitrogen oxides. So automatically greenhouse gases increasing and forest is deteriorating. So this is the negative effect. And you know, sometimes this is called the sea forest. One type, that is sea forest. In Sargasso Sea, some other that is scale life of algae, that is pheophyceans members are present. Some long vegetations of this type of algae make the forest, that is called sea forest. So that is typically not a forest, but if you assess this is a forest because 50 meters or long, that is sargassums and other typical algal members are present here. In Indian scenario, I am coming to the Indian scenario. This is called Leader Island, Swampy Forest of Sundarban, Goa, Andaman, Nicobar Island, Kerala, Maharashtra, Bhitarkanika, Odisha, etc. In your pitchy of Kerala, you can see this type of vegetation that having no ground vegetation, but some new metaphors are here, and other plants are halophytes, and some members are halophytic associates, and other members are non-halophytes. But both the halophytic and non-halophytic associates make the composition say climax one. But due to climatic conditions, you know, the oil and other heavy storms actually ruin the vegetation in some cases. So that need the monitoring. This is dry deciduous salt forest in our West Bengal, Chhattisgarh, Madhya Pradesh and Uttar Pradesh also, and this having the Surya robusta. So main composition is Surya robusta. That's why this is called sal forest. Other members are Madhuca indica, Terminalia valerica, Terminalia arjuna, Terminalia cibula, Schleicheria oleosa, Buchanania lanyon with many climbers and herbs. So I am not going to all the species compositions of a forest, but that is a forest and that having the green scenario. This is thorny forest. You know, it is a dense scrubland vegetation. Characteristics by the dry, subtropical and warm temperate areas having the rainfall 250 to 500 millimeter. And it is available in South America. You know, the common name is called Katinga. And while the South India and some northern part of Sri Lanka, this is called Deccan thorn scrub forest. Thorn forest grades into savanna woodland as the rainfalls increases, such the thorn vales of South Africa and other parts of the desert climates. So this is important. And you know, this is Eastern Himalayan forest. One is the temperate evergreen forest of oaks, many oaks. I will come to the oak plants and several species of rhododendrons, magnolias, and even that is cinnamon. The other is temperate deciduous forest dominated by maple plant, fir plant, walnut, elder, and birch. In mature evergreen forest, the trees are covered with a rich growth of mosses, ferns, and other epiphytes. Common example is pine-dominated forest in our Darjeeling area. You see, this is a good scenario of the rhododendron sanctuary in Singba area of Sikkim, North Sikkim. It is located in the Yungtham Valley of North Sikkim. First one is rhododendron dalhousie. Second one is rhododendron arboreum. After last week of April, you can see near about 40 species of different rhododendron in this rhododendron sanctuary. And a varieties of bird, a varieties of butterflies, insects, and others are available here. This is a good research site. And guras, actually red rhododendrons are regarded as guras, that is rhododendron arboreum. Some varieties are there, but rhododendron campanulatum, rhododendron Hugsoni, rhododendron falconeri, all are here. So this is the good habitat for different rhododendrons. This is magnolia. So three magnolias are here. That is Magnolia campbelli, Magnolia cathcarti, and Magnolia grandiflora. Remember, the Magnolia grandiflora included in our area from Great Britain during the British period. But now this is available naturally in different sites of the Sikkim part. And Magnolia cathcarti and Magnolia campbelli is that is common everywhere in our lower foothills of Eastern Himalaya, Darjeeling, Sikkim, and other parts. So this is Quercus. You know, this is called oak, Quercus lamellosa. That is synonym. Someone called it Cyclobelanopsis lamellosa, and other is called Catus. That is Castanopsis indica. Though other species of Castanopsis are available in different parts of the hilly areas. These are the cinnamon camphora and cinnamon giranicum. This is Dane forest. I am going to the Kerala part, Karadipara. During 2018, I have taken these pictures from Karadipara. This having the Myristica malabarica, Maduca bordoni, Bateria indica, Aglia loi, Daspirus, Candulania, Halicarna, Aromatica, Atocarpas, Hisutas, and Canarium strictum. So these are the species in your area. That is called Kattu Jati, and second one is called Chuvanna Cheralam. So first one is Myristica malavarica, wild nutmeg, and second one is Maduka borildoni, that is called Cheralam, Chuvannam Cheralam, and this species is endemic. So these are also endemic. White damask and Karakil, Vedaria and Egalia. These are Daspirus candolena, and this is called black varnished Voligarna arnotica. The second one is under the family Anacardiaceae, and this is important. Other species, that is wild jackfruit and black dhup. All these species are important because black dhup, that is Rahul dhup, is important due to presence of resin. This resin is required for varnishing the material, that is wood and wood-like materials. So this is important. And this is hill slopes of Sikkim part, eastern Himalayan adjacent to Nepal part. This forest having Maua, Malengo, Bengu and Thupi pine. You know, this is Angel Hardia spicata. This one under the family is Atlantaceae, very important family. This is Usunia Malengo bamboo. And this is the fodder plants of the red panda. So red panda use these bamboos. And even one that is younger shoot of this plants is used, and this is consumable and available in the markets of Nepal and in our West Bengal. So this is important. And another part is the Cryptomeria japonica. This plant is planted and imported from the Japan during British period. But now this is vanishing the ground flora due to acidity, increase the acidity of the soil. This is sand dunes at coastal West Bengal and Odisha part. These are the plants, you know, the pandanus, Opuntia and Ipomia pescabri, hues and binders like Spinifex lidoria at coastal part of the West Bengal. This is northeast Mizoram hills. From Mizoram University, Central University campus, you can see the Firminia colora. This is called that is fire of the forest. This is a red colored pine that is a tree available in the campus of forestry department also of Mizoram Central University. This is western Himalayan part, large pine forest. The western Himalayan broadleaf forest are divided into two forests, evergreen and deciduous forest. And evergreen broadleaf forest is dominated by oak. I am coming to the oak plants, and this is the nice picture from the Nainital hills with human habitations. And these are the oak plants. You know, Quercus semicatifolia, Quercus silicotricophora, Quercus floribunda, Quercus lanata, Quercus glauca, Quercus belluc. All are available here. And by this that is geographical picture, because this is Himalayan part, you know, and that part is central Himalaya in Nepal. This is Nepal, this is India, this part is Uttarakhand, and this having great canyons, and this is the catchment area of river Gandoki, and in Nepal that is called Narayani river, and total catchment area is near about 46,300 square kilometer. And this vast area having the different compositions of oaks and other species like Esculus indica, Jaglens regia, Carpinus viniana, Elnus nepalensis, Esar sisium, Esar acuminatum, Esar cappadocium, and Esar lobeli. So these are the figures. That is Esculus, Jaglens, this is Esculus, this is Jaglens, that is Carpinus, that is Elnus, and so on. And Elnus is important in eastern Himalayan part also. In Nepali language, this is called Utis. And these are the species of Esar. So 4 Esar species are here. That is Esar sisium, Esar acuminatum, Esar capodicum, and Esar lobeli. But if you go to the internet part, that is Esar lobeli is Esar lobelum. But lobelum is other genus, but this is mismatched with other species like lobelum. So physically all these species are same, but if you go to the floral structures and fruit that is morphology, or it may be it's germination, then different structures will come out. Other species of gymnosperms of the western Himalayan part having Araucaria, Valotaxus, Cupissus, Cycas, Gimbo, Pine, Taxus, Juniperus, Jamia, Avis, Cetrus, Piscia, Taxodium, Thuja, including Podocarpus. So all these species are here, but frequently not found in the wild condition, but in the garden of different universities and institutes, you can see all the species. So this is the Juniperus and this is the Jamia species. Now, landscape of grassland and agricultural field and rich with plantation stand of Chotnagpur Plateau. This is the lower part of the Chotnagpur Plateau, having the range of different species. You know the species available here, and some of the species are here, particularly saccharum, known as grass, and intermingled with the paddy cultivation. But all these paddy are the local varieties of paddy. So you just go to the species that is available in the region bound of this region. That is Sisu. You know, this is Dalbaria Sisu. This is Madhuka longifolia, Dacia latifolia. This is Mohul. This is Bandar Pichla. That is called Starculia urens. And this one is Anogisa latifolia. Gradually, these species are decreasing from the forest. So they are collecting the material from the other part, that is dense part of the reserve forest, and they are using these seeds for the nursery plantlets. So my question is, are these figures with similarities? If yes, percentage similarity, are these with same composition for vegetation? Are these under same forest or in same ecosystems, and so on? Questions will come from different learners or different researchers from different angles. And so these are the basic questions, and answer automatically one will answer yes, no, do not know, maybe, need study, need research, need survey, need monitoring, need linkage, not my duty. So I can say that is a research work, and this is a practice based on the research, and need a linkage locally or regionally, and after that nationally. After that you just contribute your research part to make the big research data. So now I will come to the vegetation. So you know, all of you know that is floral compositions with various species is called floral interactions, and this is a important parameter for studying the ecosystem health. So important criteria used during classification of vegetation based on physiognomy, structure, function, composition, and dynamics. What is dynamics? Succession and final changes, habitat, its relationship, and history also. So comments by the earlier workers further found that structures, the functions are the most satisfactory criteria for classifying the vegetation. But Champion, his classifications, he gave the first complete classifications of vegetation of India based on the physiognomy characters. But now, 5 types of natural vegetation found in India, which are tropical evergreen forest, tropical deciduous forest, tropical thorn forest, mountain forest, and mangrove forest. But if you go to the Champion and says classifications, or Purian Veromi's classifications, very different subtypes will come here. So don't go to the confused part of this Champion and says classifications. You take the 5 types for your best research. This is tropical evergreen forest that is found in case of Andaman and Nicobar Island, which comes up, which trees of the Arabian Sea, the coastlines of Peninsular India, and the greater Assam regions of the Northeast India. Small remnants of evergreen forest are found in Odisha. If you go to the Similipal Biosphere Reserve, then you can see the compositions of the Western Ghats as a whole, including Eastern Ghat part, flora and fauna. And then tropical deciduous forest. This is Madhya Pradesh, Uttar Pradesh, Odisha, Chhattisgarh, Jharkhand, and some other parts of the Maharashtra. This is tropical thorn forest that I mentioned earlier. That is Gujarat part, and little run of Kutch, including Rajasthan part. Characterized by some tropical and subtropical forest tree species, Salvadora and other species. And you know that is the composition change their pattern from time to time. Mountain forest. This forest can be found in the higher hilly areas of Kerala and Tamil Nadu part, also in the regions of the eastern Himalayan parts. You know your area, including Assam part, West Bengal part, Sikkim part, and Arunachal Pradesh up to Nagaland. So now I will come to the mangrove forest part. This is a picture that is Godavari, Krishna, Kaveri basins and Mahananda deltas. This forest having different compositions, and this forest is called littoral and swampy forest because high tide and low tide are the characteristics of this vegetation. Now I will come to the species compositions in mangrove forest. You know, this is Xylocarpus, this is called Pasur, this is Brogoria, this is Rhizophora, this is Heritiera. Depending upon this Heritiera or Sundari, the name is Sundarban. Now this is Nipa palm and mangrove date palm. This is Nipa palm. This is called Nipa palm or Gol Pata, and this is called that is mangrove date palm. All are diminishing from the forest day by day. This is grey mangrove and Indian mangrove. All these species are Avicennia marina and Semiopterale. So, cumulative associations and green patch with plant cover is called vegetation. Not out. That is this is a contradictory part. That it may be greater vegetations or lesser vegetation, but in all that is vegetations. Though it is a species composition, but its characteristic can change from time to time for its impact factor, that is topography and climatic condition, from time to time. So important parameters that to be included in the research, that is topography, climate, graphic of soil factor, body factor and site factors. What is site factors? You can see various anthropogenic activities imposed on the local vegetations, local flora, local fauna, and so on. These factors are called site factors. This is a species and this is a composition, and that is dominated by silver fir, Grevillea robusta. This is the picture taken from Nainital Park. This is a diverse forest that having the ordinations, but in a lower Kudil area, the composition is different. This is called Windelandlia. This is a species of Rubiaceae. So, kilometers apart from the upper part of the hilly tract, the composition is different. This is a specific composition. This is a specific composition dominated by two species. So, this is a composition that having the plantation crop, that is wheat cultivation along with other tree species, that is eucalyptus. So this is different habitats and different composition. This is vegetation, flora, fauna, compositions of soil, microbes, all are different. That is lower part of the Himalayan tract having no human habitations and no human interactions. So gradually the forest cover is increasing and the health is good. So this is mixed, that is population. This population is Colocasia population. This is the plantation coconut. This is other vegetables. This is rice, that is hilly rice, and some local populations are here. So these are the different populations: human populations, animal populations, plant population, plantation populations, and even agricultural introduced species populations. My discussions depends upon the vegetation study of forest, cottage, biomass, different species, compositions, ecosystems, ordination, discussion, field trial, publications, post-publication, review, research and interventions, even there's comparative account and basic applications. So, as a whole, now we'll come to the vegetations. What that is recognized as a cover flora up on ground that change its characters time to time due to change of season. Therefore, vegetation is a general term for the plant life of a region. It refers to the ground cover provided by plants and is by far the most abundant biotic elements of the biosphere. So you see different habitats from different times, and the parameters is flora, study of the vegetation, study of the diversity, study of the comparative aspects of vegetation in nearby to varied ecosystems. That is called beta diversity. This may be forest, grassland, degraded lands and plantation stands. Why forest conservation? So I will not come to these points because deforestation process to prevent it, balance the nature, breeding stock and so on, that is required. Causes of forest depletion, all these are the causes necessary for conservation. This is all are the field parameters. Continuously, that is disease resistance, high productivity, high ecological amplitude, high fitness index, low mortality rate, high flexibility, economic value, yield timber, firewood, poles, bowls, fruits, seeds, barks, roots from plants, even from animals, ivory, horns, fur, scales, meat, feathers, all will come to the forest. All will come to the different habitats from one place to another place, or it may be from one side to another side. And even from the forest, soil, microflora, different types of fungi, bacteria, all are there. So many soil-borne myxomycetes and actinomycetes are the source of antibiotics and similar products in laboratory. Various mushrooms, small medicinal herbs, all are important. And ultimately recreation, you can see the forest, you can see the solar forest of your area, you can see the thorny forest of Gujarat and Gir, you can see the evergreen forest of Andaman, you can see the little and swampy forest of Sundarban. You can go to the eastern Himalayan part of Darjeeling in West Bengal, Sikkim, Arunachal Pradesh and Nagaland. the western Himalayan part of the Bengal, Nainital, Almora, etc. for your study and research. So this is not only education but also research and extension. And now obviously we will go to the endangered species. Need to study, need to record, need to data, and after that you can critically analyze endangered, endemic, threatened, different species are there, and the situation, the ecological amplitude, ecological habitat, ecological status, that is required. So some species of the animals, some species of plants, all are important, but need to know the condition that all are wild, all are that is cultivated, all are it may be mixed one, or it may be the alien species. So that is required. And before going to the field, you need to know the parameter. These are the threatened species from I think Kerala, Uttar Pradesh, Daman and Diu, Western Ghat, Jammu Kashmir, Tamil Nadu, Jammu and Kashmir, and Karnataka. These are the threatened species. You can take actions on one species or more than one species, and you can take steps to study these species. You can regenerate, you can propagate, you can go to the tissue culture techniques on those plants. Methods of conservation are different. You know, that is knowledge on the forest and its composition, knowledge on wildlife, appointments of official, project lodge, different activities, different acts, and others. So all the parameters need study. And educational research activities, that is also the focus points of this research. But in case of the research of ecological study, need the methodology. So you take any kinds of quadrant, different that is shape, that is area fixed for the quadrant. It may be round, it may be triangular, quadrangular, rectangular, pentagonal, the other kind. You just take the minimum size, and after that increase continuously. If the number is fixed, then you just fix up the quadrant. Best quadrant is the quadrangular quadrant, and this is common type. This is quadrangular quadrant in which the species is here. You count the number of the species, you count the diameter of the species, you record the diameter, breast height, and canopy cover, and species composition. But you need to know, in case of the plain forest, you can take the decision that plain forest, that I will take the quadrant study. This is that is slopey land, so need the aspect, not exposure. So you take the line transit method. So quadrant study for 10 into 10 meter for tree species, 5 into 5 meter for shrubs, and 1 meter for herbs. So this is the quadrant size recommended for the study. Similarly, you can take the cryptogams, seaweeds, grasses, large heathers, scrubs, woodland canopies, ecosystems, herbs of lateritic forest, herbs of your area, shrubs of lateritic forest, tree species of same site or different sites, locally, nationally, or globally. And now we can study through remote sensing or geosatellite image processing, that is methodology. But that is critical. But if you are within your home, so you will start your research from your home and then continue it to the locally, and then nationally, then go to globally. Then quadrant size, you know, small elements will cover that is less than 50%. Small animals where cover is 50% and less than that is 0.1 meter into 0.1 meter. And large animals, ants nest, termitarium, and even termitarium of our area, the specific selected shape of the quadrant size is fixed for study. So this is plain land, but exposure and aspect. In case of hills, you just take the line transit, 30 meters or 50 meters, whatever it may be, depending upon the types of the slopes. These are the hill slopes. You know the hill slopes, and all the species are scattered, but a few species within the transit line. So record the transit line species and then record the diameter and canopy cover. So physiognomic character-based species composition study is essential because seed after germination going to seedling stage, and after that 1 to 3 meter, that is sapling, and 3 to 7 meter, that is pole, 7 to 10 meter, that is bole, and above 10 meter, that is tree species. So you need to study these parameters, and these are the species that you can take for the physiognomic study. But you know that what condition prevails within the forest. That same species, Surya robusta, in herbaceous condition, in shrubby condition, or tree condition. So recording of data is important. It may be secondary data, it may be primary data, but in photography in field, photography that is available from the research articles. In soil also, degradation mark, that is cut points of different poles or trees, even shrubs. Side factors, videography of the landscape, it may be portrait or it may be landscape, sound of any, noise of any, dialect of local people, sacred group study, forest official and their positions, even forest office landmarks. That is forest office, that is forest office created landmark by stamp and so on. And DBH, diameter at breast height, because a fixed diameter will be taken from the species if the height of the choosable part of the tree is same, then 4 feet 6 inches or 1.37 meter is taken for this study. In guard of all tall trees, you can take guard at 1.37 meter, and this is coined by BCFT, British Commonwealth Forest Terminology. And similarly, from this you can take the frequency, density, abundance. From biomass, you can assess the relative density and relative abundance. And similarly, soil parameters, you can study soil, carbon, soil organic matter, nitrogen, phosphate, potash, calcium, etc. and also water holding capacity to correlate the vegetation in a site-specific study. This is a height at diameter at breast height. So people are taking diameter at that level. This is called 1.37 meter. This is girth. He is taking girth, and after that he will calculate the total log size. Here that is taking girth at DBH. So this is a soil vegetation, and this is called dry deciduous soil vegetation. And this one having that is a dense forest of the Kerala. You can take the girth, or so that is height of the diameter from this part. And you can use the tape, or you can use the general law, that is 2 feet, that is 612 centimeter, 2.5 feet, that is 76 centimeter, or 4 feet, that is 122 centimeter, and so on, by hand or by foot, and so on. Consider girth for species on and from 10 centimeter. That is not required. So felling and silviculture. You know, silviculture is a process by which forest crops are tended, removed, and replaced by new one. This is called silviculture. So silvicultural technique recommends you just consider the species in which 40 centimeter GBH is available. But in an area, 80% species under the above 40 centimeter cm girth, so it will be considered for the felling. If it is a clearly basis, then that is called clear felling. If it is a strip basis, that is strip felling, and that is depending upon the species characteristics also. Say for example, if it the coppice species, then after clearly felled from the area, automatically they will regenerate. That is called Surya robusta and eucalyptus species. All are the coppice species. So these are the coppice power producing species. No coppice, having nothing, after cutting, no one will regenerate. So this is that is no coppice species. Acacia auriculiformis. And bad coppice, having bad shoot production, but not original shoot production. That is in case of Bombex Siva. Many species are there. So biological spectrum study is important. You just study the cryptophytes, hemiphytes, therophytes, and hemicryptophytes, even phanerophytes. And composition study, and you can assess the previous data and present data, and then can assess the health of the ecosystem. Threats, you know, different threats are there. That is agriculture, grazing, scrapping, deforestation, fire, pollution, fragmentation, introductions of exotic weeds like lantana, camera in your area, Parthenium, Histonopolis in your area. So consider these species, if introduced within the same land, automatically quality of the forest is going to be deteriorated. So need special attention. Here is that is level-based study, that is alpha, beta, and gamma. So if you study from a vegetation of your university campus or college campus, or from your nearby forest, that is alpha diversity. If you compare between the two stands or two compositions, this is called beta diversity. If you consider whole as a, that is called the gamma diversity. It may be the diversity of large scale, that is geographical land. It may be biosphere, or it may be any country species. So this is quantitative features of the community of the vegetation study in the forest. So need to study phanerophytes. So we can consider tree more than 30 meter tall and trees between 8 to 30 meter tall. And another category that is 2 to 8 meter high. You just elicit these tree species, even shrubs that is 2 meter high. So you just consider 30 meter tall and 8 to 30 meter tall, two types. Similarly, chemophytes, similarly for hemicryptophytes, another type cryptophytes and therophytes. Consider all the types and then study. Why do we study vegetation? Because you know the biogeochemical cycles throughout the year is going on, and different components available in the ecosphere, and they are coming from the soil, from the water, from the aquatic bodies, from the forest, from the grassland, from your area. So need to study. So mostly those elements which are required for all organisms and their recycling is required. So need study in atmosphere, in hydrosphere, in mesosphere, in thermosphere. So it can regulate the process and important parameters that can balance the water cycle, carbon cycle, nitrogen cycle, phosphorus cycles, and so on. It is also of great importance in local and global energy balance. Now it regulates the greenhouse gases and takes functions to mitigate the level of the greenhouse. So need study. Direct and indirect impact of vegetation. You know, mitigate the climate change on fire regime, impact of elements on vegetation, coverage of micro and macro elements, saver of micro elements, microflora in and around the vegetation. Why? On paper published in the PLOS journal, they showed that complexity in African savanna, direct, indirect, and cascading effects of the animal diversity, animal density, rainfall, and vegetation availability. Paper showed cross-PM modeling using mammals and vegetation data from 3 different wildlife sanctuaries and reserves in South KwaZulu Natal. Because you know, if heavy dominant tree species are there, so least number of the ground vegetation automatically will not allow to graze the elephants. So heavy tree vegetation that will they will eliminate the forage of the wild animals like wild buffaloes and wild elephants. So positively, if the lesser number of trees, then automatically will come more and more grass species. So it will attract the grazers and browsers. So this is important. So now the result and conclusion part. This is frequency. You know everyone that is required total number of quadrate in which species occur by total number of quadrate studies is called frequency. Density. This is number of individual of a species by total number of quadrate studies. And this is a table. In such a table, you can study in a field and then you just assess the density, abundance, that is density, abundance, and frequency. But obviously, we will consider that is 100 for density, 100 for abundance, and 100 for frequency. And cumulatively, if you that is add, that is important value index will come, and the cumulative value is 100 plus 100 plus 100 is equal to 300. So high frequency and low abundance means the regular distributions of species. Low frequency and high abundance mean contagious distributions of species. So this is dominance indices. So different indices are here. That is Simpson's index. That is called Simpson's diversity index. From your important value index, you can assess that is Simpson's index or Simpson's diversity index, because this is the reciprocal part, that is 1 minus diversity index, and this is the diversity index, number of individual per each species, total number of all individuals. So you can consider it may be 0 or it may be 1. Within 1, you can consider 1 minus that is value. So Simpson's index of diversity is 1 minus d, because parametric data will allow to develop graph, but to know more dominant graph, you have to go to the Simpson's index of reciprocality. So that is reciprocal index, that is 1 by d. And similarly, examples in case of different species of dry deciduous forest, you can assess that is species wise in minus 1 number and serial number, and putting these values into Simpson's index, you can get the Simpson's index of diversity, that is 0.7, and Simpson's reciprocal index is 3.33. So that is better to draw the line. And biomass, You know, that is cumulative production is called biomass, but in a dry oil basis, you have to study the green biomass. After that, you have to transfer the same in the dry biomass. You can consider above ground biomass and underground biomass, and ultimately you can study whole biomass. So biomass calculation is the method that is dry biomass of 25 centimeter into 25 centimeter, that is 25 centimeter square area by 625 into 100 into 100 gram per meter square. So in this way you can calculate average basal area using this formula. So this is a table. Through this table you can calculate the basal area. This is important value index. Already I have mentioned IA is equal to RA plus RD plus RA. R stands for relativeness. So this is totality of the important value index values, that is frequency, density, and abundance. And automatically it will be 300. If it is more, then reduce the value. So different formulae: diversity index, dominance index or concentration index, evenness index, richness index, and similarity index. So you can use the similarity of the species of different strands. That is index of similarity is equal to 2 into C by A plus B into 100. C is equal to common species that is available both in the A stand and B stand, and A is the total number of species, B is the total number of species found in B stand. So abstract community is a community in which tree species is dominant, shrub is dominant, and herb is dominant. So after study, you just make the abstract community, one from tree, another from shrub, another from herb. So these are the abstract community. So if you study in seasonal way, then you can get the same parameter for tree species, but in case of shrubby and herbaceous species, it will change. So seasonal influence and seasonal study is essential to know the status, and it will be continued year after year. What is evenness of species? The natural logarithm of species richness is called evenness. So it is another parameter. So you can take the non-timber forestry species. That is primary productivity, secondary productivity, and secondary products is called NTAPS, but now that is not non-timber forest produces. It is called major product also, because fruits, flower, leaves, medicinal plant parts, the whole heart, seeds, all are used widely. And if you consider in terms of the money table, then that is more vigorous than that of the yield, that is wood product. How to start your study? Field study is essential. Random sampling is essential. GPS reading latitude, longitude, altitude is essential. But do not write the latitude, longitude, and altitude, because if you place the altitude, latitude, and longitude and I think record the species of endangered or threatened category, another will go to the field and will collect the same species. So this is another negative effect on the GPS reading. So obviously you take the GPS reading, but do not place the GPS reading along with the I think threatened species. Make a local map with the help of the local people using participatory real appraisal. Here a technique is essential. Study the characters of vegetation, soil, flora, fauna, soil microbes to know the condition of ecosystems on trial basis. And ultimately you make the important value index and then different indices. So now conclusion, I conclude that go to the field with full basic knowledge and apply the methodology for proper assessment of vegetation by knowing present scenario of ecosystem. Based on vegetation, characters in the field with prior way, not the earlier knowledge established too. So it is your work. Continue and repeat it seasonally, even year after year. Then compare the status. Make a line of point value. Assess the quality that gradually increasing or decreasing. Start whole to part, not from part to whole. This is my I think request. Go to the whole part and start from whole part to the part, not from part to whole. Upload your data on global platform and publish your data for other use. Remember, your work not for yours, it's for all. These are the bibliography for study, and there is some photographs for that study. That is carbidarium study in soil vegetation, species study. That is degraded forestland study in monsoon. Thanks to all. I acknowledge Managing Trustee Avinashilingam Institute for Home Science and Higher Education for Women, Tamil Nadu, the Honorable Chancellor Sir, the Honorable Ma'am, the Honorable Vice Chancellor Ma'am, Registrar Ma'am, Dean School of Bioscience, Coordinator, UGC Stride Project, all the faculty members, Department of Botany, Zoology, Biochemistry, Biotechnology and Bioinformatics, and Physician Assistant, and my college officer in charge, principal, chief conservator of forest, conservator of forest, DFOs, range officer, beat officer, joint forest management committee members, scholars, students of PG department of my college, your institute and others college that are I think today present as participants, my students. Thanks to all. Have a nice day.
Thank you, sir.
Thank you, sir, for your brief explanation, and this was informative and thought-provoking session. Thank you, sir. Now the forum is open for discussion. If you have any doubts, please post in chat box or now the forum is open for discussion. If anyone have any doubts, please post
Yes,
in
madam.
chat box or I request to unmute yourself and ask the doubt itself.
That is confined not for this day. You can continue and repeatedly you can throw your I think problem and your I think query to me directly through mail or directly call over phone or through institute I think organizers today's program. I'm always ready to share my views.
So there's a question in the chat box. Which vegetation needs lot of water to grow? Can't it be avoided in water body?
Madam, again.
So
Again,
which
the-
vegetation needs lot of water to grow?
Actually, this is a good question, but basically, some aquatic bodies are available in the aquatic ecosystem. It may be the freshwater ecosystem, it may be the brackish or marine ecosystem, but we have to know the situation under which it is working. It may be, I think, in nutrient-rich vegetation. So automatically, different nutrients are coming from the other areas, that is rainfed areas, to the water bodies, and then successively the situation is not that is fixed. I already mentioned earlier, the vegetation is always dynamic. Nature is unique. Vegetation is unique because nothing is fixed there. So once it will under climax condition, that is fixed, but immediately one will dominate in a situation and other will act as a co-dominant. Say for example, if one species is dominant in one area, then other accordingly, and they will deteriorate the vegetation. You lost to the connection.
Thank you, sir. Raji?
If there is no more doubts, we shall move on to break. Now we'll have a short break and please join us at 11:40 for the next technical section.
Participants, you need not log off. You can stay online. The next speaker will join immediately. Good morning, ma'am. Pampi ma'am has joined the meet.
Yes, ma'am, join.
Yes, ma'am. Yes, ma'am. Ma'am, can we start?
Oh, 2 minutes, 2 minutes, sir.
Yes, ma'am. Yes, ma'am. We'll wait.
Follow? Yes, ma'am, you can start.
Okay, ma'am.
Madam,
Now, we will begin the next technical session by introducing the resource person, Dr. Pampikosh. She is an assistant professor of botany, Shivabharati Mahavidyalaya, Katpagari, Jargram, West Bengal since 2008 till date. She served as assistant teacher in botany from 1999 to 2007 at various government-aided schools at West Bengal, appointed by West Bengal School Service Commission. She acted as NSS program officer and conducted various programs like NSS Winter Camp and Environmental Field Study. She has completed two minor research projects funded by UGC ERO, Kolkata, on ethnobotany. She has been published 37 research papers in reputed scientific national and international journals. She was a co-author of a textbook in botany, namely Prayog Viktik Banaspati Vidya, and other book chapters written by her since 2008. She is editor of a recently published book, Fundamentals on Plant and Soil Microbial Interactions, for different books published from different organizations. Her interest is on ethnobotany, plant taxonomy, soil AM fungi, experimental plant physiology with biofertilizers, medicinal plants, soil-borne fungi, socio-cultural aspects of marginalized people of West Bengal, Odisha and Jharkhand. She delivered two national-level invited talks this year organized by College Authority from Maharashtra and Madhya Pradesh. Dr. Ghosh is not only actively participated in science research activities, but she is doing work in social science research, acted as District Sustainability Mentor for Jhargram District under NGNCRE, Department of Higher Education, Ministry of Education, Government of India. We are fortunate for having you, ma'am. The session is over to you.
Thank you. Thank you for introducing me. Good morning to all present here. Think you are able to see my PPT.
Yes, ma'am.
Okay. So, good morning to one and all present here. My topic is of fungal biofertilizer and its application in agriculture and forestry. I am happy to be here in this 6 days long online training program on environmental sustainability and research ethics sponsored by UGC scheme for transdisciplinary research for India's developing economy. I think this is the fourth day. I am the second speaker of the first session. I am thankful to all the organizing committee members of the botany, zoology, biochemistry, biotechnology and bioinformatics and physical assistant departments of the AIHSHEW deemed to be University Coimbatore, Tamil Nadu, to giving me opportunity to deliver a lecture emphasizing on sustainability in the area of agriculture and environment. As my topic is fungal biofertilizer and its application in agriculture and forestry, first I want to start my lecture with a brief introduction about biofertilizer. Actually, everyone is familiar with the term fertilizer, and now biofertilizer also the common term. Fertilizer is any material and natural or synthetic origin that enhance plants growth by supplying nutrients to the plant tissue when applied to the soil where the plant is inhabiting. Every living being needs nutrients for its own growth and reproduction. We also need nutrient for our body and mental health, as plants are preparing their food by own, so they need ingredients for the preparation of their food, and these can be taken from soil only without human intervention, as we are totally dependent on plants for every second, knowingly or unknowingly. We appreciate only productive plants and need more and more production. That's why we are using fertilizers, maybe chemical or organic, but our aim should be maintaining proper soil health for the sustainability of our environment in every respect. So the term biofertilizer refers to preparation containing live microbes which helps in enhancing the soil fertility either by fixing atmospheric nitrogen, solubilization of phosphorus, or decomposing organic wastes, or by augmenting plant growth by producing growth hormones with their biological activities. As our air is full of nitrogen, but microorganisms can only fix them in the form of ammonia or nitrates or nitrites, which plants can absorb through their root hairs for their use. And from roots, it is transforming to us. Approximately 30 to 65% of total soil phosphorus is in organic form, but are not usually available to the residing plants. Microorganisms like bacteria are the main phosphate solubilizer, which solubilize the insoluble form of phosphate like tricalcium, iron, or aluminium phosphate into the soluble form. Fungi again decompose the dead bodies of the plants and animals, thus help in recycling the minerals in the abiotic and biotic elements of the environment. Actually, fungi also help in mobilizing phosphorus to the host plants. Biofertilizers directly improve plant growth by producing plant growth regulators such as auxin, gibberellins, cytokinins, etc. So, according to Bhesi, biofertilizers can be defined as biological products containing living microorganisms that, when applied to seeds, plant surfaces, or soil, promote growth by several mechanisms such as increasing root biomass or root area and increasing nutrient uptake capacity of the plants. So, yes, ma'am.
The slide was not moving actually. Now it has
Oh,
come to
slide is
importance.
moving. Okay,
Yes,
okay,
ma'am.
okay.
Now it has come to importance of biofertilizer.
Oh, it comes. Okay, okay. Importance,
Yes, ma'am. Yes, ma'am.
Okay, okay, okay. Moving. Slide
Now it is in the third slide, ma'am.
Huh?
Third slide. Now it is in the third slide.
Slide is moving. Slide is able to.
Now
You
it
can
is
see?
visible. Yes, ma'am. Now it is
Okay, okay.
visible. It is in the third slide.
Okay.
Okay.
Now, importance of biofertilizer or role of biofertilizer. The biofertilizer help to get high yield of crops by making the soil rich with nutrients and increase useful microorganisms necessary for the growth of the plants. Plant growth can be increased if biofertilizers are used because they contain natural components which do not harm the plants but do the vice versa. Biofertilizers promotes the development of mycorrhizal associations which increase the availability of phosphorus of the soil. Biofertilizers destroy those harmful components previously, as I said, from the soil which cause disease in the plants. Plants can also be protected against drought and other strict conditions by using biofertilizers like M. fungi or VAM fungi. Biofertilizers are cost effective, even poor farmers can make use of them. They are environment friendly and protect the environment against pollutants. They are effective alternatives to the hazardous synthetic fertilizers. So, but as short-term crops require rapid supply of nutrients to produce high yield, chemical fertilizer application is essential, which led to environmental pollution and health problems like chronic kidney disease, cardiovascular disease, cancer, blue baby syndrome, according to report. So, mitigate the situation, Sri Lankan government decided to develop and use biofertilizers using its own soil microbial biodiversity in 2019. Sri Lankan president that time promised in this election campaign for transition of the country's farmers to organic agriculture over a period of 10 years. Next, he imposed a nationwide ban on the importation and use of synthetic fertilizers and pesticide and ordering the country's 2 million farmers to go organic. The result was very brutal. Rice production fell 20% in just the first 6 months and forced to import $450 million worth of rice. Not only rice, it also decreased the tree production. So it is not possible to be organic or using biofertilizers in totality for all crops. To develop the present soil health and also sustain the crop productivity, we have to use biofertilizers on 30% basis preliminary. It will take time and take time for be total organic. Technology based real research must be encouraged to get real result before application of biofertilizer in the field. I am going to next slide. Now, classification of biofertilizers, producing organisms. Here you can see symbiotic, associative and free living organisms, and they help in nitrogen fixation, phosphate fixation, organic matter decomposition, and in below line for the place inconvenience, I wrote here that potassium solubilizing bacteria and PGPR. Production of now in this classification of biofertilizer producing microorganisms on the basis of their association and production of ultimate compound which is easy to be absorbed by the plants for their growth environment. First, nitrogen fixture. Here rhizobium is rhizobium in soil. Actually, it is inhibiting in every soil, but its number may vary due to soil health and its host plant. And these are symbiotic to legume roots. They fixes atmospheric nitrogen into usable form, that is ammonia for the plant. These microorganisms are important for legumes and oilseeds. Again, Frankia, when it comes in non-legume, fixes considerable amount of nitrogen when present in the root of non-legume plants as symbiont. Frankia present in the roots of Alnus, Casuarina and many more plants. And excellent example for the benefits they provide to the ecosystem by way of nitrogen inputs. Then another symbiotic microorganisms like cyanobacteria present in the environment in symbiotic association with azolla fern, cycas roots and many other plant roots and fixes nitrogen for their own and hosts. Now we are trying to mimic their ability and azolla, a free-floating water fern which contain Avina azulli in their leaf, is used as biofertilizer for wetland rice cultivation, and it obviously show good results. Then come the associative microorganisms, that is bacteria, is typical for cereal and free-living nitrogen-fixing bacteria that may adhere to the host roots. For example, written as Azospirillum, Beijeringia, Daxia, etc. These associative nitrogen fixer convert dinitrogen gas into ammonia in casual association with plant, hence enhance the amount of nitrogen within the associative cereals or grasses. Then come to free-living non-symbiotic bacteria like Azotobacter, Clostridium, Astrobacter. They are present in the soil which increase soil nitrogen by its own property of nitrogen fixation. Experiments with Azotobacter culture and crop plant at the IARI New Delhi led us to believe that significant increase in growth and yield of wheat, rice and vegetable crops could be obtained in pot culture. The population of Azotobacter in the rhizosphere of crop plants and in uncultivated soil is generally low. To overcome this repeated application of Azotobacter during different stages of growth of a crop is now being recommended with the object of increasing the number of bacteria in soil and enhance the soil health, better health. Now come to the next point, the phosphate fixers. Phosphate fixers are divided into two, that is phosphate solubilizers and phosphate mobilizers. Phosphate is a very important mineral for plants. Here are some bacteria like Bacillus, Pseudomonas, and some fungi like in a red color, Aspergillus, Penicillium are the most phosphate solubilizing organisms. They solubilize mineral phosphate by releasing low molecular weight organic acids such as formic, acetic, propionic, lactic, etc. and acid phosphatases like phytage. So phosphate mobilizing, so they are phosphate solubilizing, and let's come to phosphate mobilizers. Phosphate mobilizing microbes mobilize phosphorus by changing pH of the soil and also by producing chelating substances. Actually, in this case, phosphate mobilizers, mycorrhizae are most well-known biofertilizer, and thus they help in mobilization of insoluble and fixed form of phosphorus in the soil. So M like glomas, zygospora, acalospora and other, and ECM and ectomycorrhizae are good phosphate mobilizers. The key difference between phosphate solubilizing and phosphate mobilizing is PSM hydrolyze organic and inorganic insoluble phosphorus compounds to soluble phosphorus, while phosphate mobilizing microorganisms mobilize insoluble and fixed form of phosphorus in the soil through solubilization and mineralization. Plant absorb phosphorus in the form of orthophosphate, and phosphorus is the second most applied fertilizer in the agricultural field. Next, come to potassium solubilizing bacteria are Bacillus edapicus, Bacillus mucilaginous. They try to help solubilize potassium, and they help the plants to get it. Then another group of biofertilizer producing organisms are organic matter decomposers. Soil organic matter comprises residues of plant and animals at all stages of decomposition mediated by soil microorganisms. Here you can see cellulitic microorganisms as trichoderma, and many more are penicillium, aspergillus, and all are fungus. And lignolytic fungi are mainly written here are Polyporus, Agaricus, Ganoderma. Basically, they are under Basidiomycota. And organic matter is essential for the formation of soil aggregation, soil structure, which ultimately determines the extent of soil aeration and rooting habit of plants. Next, come to PGPR, plant growth promoting rhizobacteria, as one of the active ingredients in biofertilizer formulation. They live inside plant and exchange metabolites with them directly, hence improve their growth and yield. They also suppress phytopathogens by producing chemicals that inhibit the growth of plant pathogens. They also produce phytohormones also. So here the important steps in relation to production of biofertilizers. First, we have to isolate the organisms which we want to prepare as a biofertilizer, and take initiative to isolate these organisms from soil or root or water. Then next is media preparation, then inoculation and mass culture. Lastly, carrier. This is a very important thing, that there should be a very ideal carrier should be there, because this is the last step. Ideal carrier material is necessary for the production of good quality of biofertilizer actually. and they may be peat soil, lignite, charcoal, sand soil mixture. And ideal carrier material should be cheaper in cost, locally available, non-toxic, and without any chemical, and easy to process. Next, isolation method, and I think most of you know this. And these are there are different methods. Serial dilution method for bacteria and fungi from soil and water, and when we are trying to isolate bacteria from root nodules, we collect the nodules and isolate the bacteria from this root nodule, and we have sterile these root nodules should be sterilized with the sterile materials. And one thing is written wrong here, ethanol. Then pressing is required. After pressing, the material has to be transferred to the desired nutrient medium for colony growth. And for M biofertilizer production, wet sieving and decanting method is needed to isolate the M spores from the soil for culture. Next, isolation of different biofertilizer inoculant. Previously, I said it. Next, these are the biofertilizer organisms. I am trying to show you that other organisms like Rhizobium, Azotobacter, PSB, blue-green algae, Azoospermum, they are very small in context to VA mycorrhizal spores. So, VA mycorrhizal spores, handling VA mycorrhizal spores is more easily, and to handle them and making biofertilizers from them is more easy method, because they need they are cost effective and need low cost, and they don't need any chemical media, the agar media. They need only sand and soil, and as they are bigger in size, they can be easily handled. Next, here there are several fungal components. Here again spelling mistake, used as biofertilizers like Fusarium solani, Trichoderma, Azoospermum, Penicillium, Chitomium, Glyocladium, and so on. And each of these fungal components have different activities as biofertilizer. As Fusarium enrich compost biofertilizer, degrade plant residues, and maintain carbon nitrogen balance in the soil. Trichoderma Hargianum, Aspergillus penicillium together enhance compost degradation. Aspergillus niger solubilizing unavailable phosphate for plant use through the secretion of organic acid and improve soil quality. And again, many species of penicillium, they supply essential minerals to the plants and induce abiotic stress resistance to the young seedlings and protect plants from fungal pathogen. And lastly, Glomas, Zygospora, Acalospora, Sclerosystem, Enterophosphora are used as M fungi, and they forming bridge between root of the host plant and soil. Again, here is a small mistake, sorry for this. Hence, a help in capturing nutrients like several micro and macronutrients to the host plants. And M fungi excrete the gluey, sugary substance, glomalin, which help in soil aggregation and also increase the soil health quality. Next, I come to mycorrhiza, and I want to emphasize on this topic. So from the previous slide, I can say that microorganisms such as bacteria, actinomycetes, BGN, fungi are the sole component of producing biofertilizer, which is essential for the sustainability of soil and environment. I actually want to emphasize on mycorrhizal biofertilizer. And here you can see two different type of mycorrhizae, ectomycorrhizae and endomycorrhizae. Mycorrhizae actually is a symbiotic association composed of fungus and roots of higher plant. In a mycorrhizal association, the fungus colonizes the host plant rootlets or roots, either intracellularly as in arbuscular mycorrhizal fungi or extracellularly as in ectomycorrhizal fungi, and here rootlets are not present in the host plants. These fungi cannot be grown in pure culture but must be grown in association with plant roots. They form branch structure called arbuscules within the, you can see here, arbuscules and vesicles grown in the association with the plant roots, and these are the branch structure arbuscules, and thus they are known as arbuscular mycorrhizae. And these are the vesicles, balloon-like structure within the host cortex, host plants root cortex, and these are actually the site of nutrient exchange between the fungus and the host. In case of ectomycorrhizae, they form seed outside the root, and from that point they enter the cortex and remain only in the outer cortical cell to form a network called as Hartig net, which is essential part of the symbiotic structure. All the nutrients are absorbed by the fungal mantle and transferred to the root through the Hartig net. And now, M. fungi actually occur in root of most angiosperm, pteridophytes, bryophytes, although absent in the plants which form only ectomycorrhizae. Hence, I am trying to. This is during my research, I have studied 144 plant species and observed that most of the plants, about 95% plants, monocot, dicot, and fern, they are mycorrhizal. Now, role of mycorrhizae in sustainable agro practice. M. fungi are ubiquitous and symbiotic, live in most agricultural crops except Brassicaceae, and have the ability to mobilize mineral nutrients from the soil to their host plants. M. fungi, by means of their hyphal growth within the soil, extend the volume of soil, which can be accessed by plant roots for uptake and movement of water and nutrients. M. fungi again improve soil structure by the production of bromelain, previously I said also, and control host plant diseases, thus helps host plants to grow better. M. fungi convert insoluble soil phosphate into soluble form through the secretion of organic acids with concomitant change in texture, structure, and water holding capacity of the soil. So, recently many countries are trying to use these M biofertilizers, and according to report, USA, Bangladesh, India, Egypt, Nigeria, they are applying these M biofertilizers on several crops like rice, soybean, stevia, and sweet fennel, etc. And this is during my PhD research for production of M biofertilizer, application of M biofertilizer on two different medicinal plant. Actually, I have not done my work on agricultural crop because my study on medicinal plant, M host and medicinal plant. So I took two medicinal plants widely present here, and these two plants are peduncle and kalanchoe. During this time, actually in the previous slide I showed that sterilized garden soil was taken in every pot, 2 kg per pot, and 30 gram inocula, and inocula was prepared from Glomus mossi, Zygospora margarita, and Acaspora leavis. They are present in this soil, that's why they are taken. And now I want to show you how M. fungi are good as biofertilizer from my study result. Actually, I have been studied on VAM inoculation effect on various parameters like root and shoot length, leaf area and leaf number, root and shoot diameter, above ground biomass, root volume, underground biomass, and again chlorophyll content of the leaf, carbohydrate content of the leaf, and total protein content of the leaves. This study was performed for the period of 6 months, and after 3 months, undisturbed and well-managed watering and caring data were collected on fourth, fifth, on third, fourth, fifth, and sixth month consecutively, and data were analyzed statistically. And this is the result. From the result and from this data, I have observed that the length of the shoot in 3 different M inoculated, that is 3 different means Glomasmasi, Zygospora margarita, and Acalospora levis inoculated plant on Kalanchopinata and Cathanthus roseus, that they increased the shoot length considerably in all 4 harvesting period. That means after 3rd, 4th, 5th, and 6th month. The result not significant over their control counterpart in all the cases in Kalanchopinata, but it is significant in Cathanthus roseus. Later I will show you how dependent these 2 plants on the M biofertilizer. Highest percentage of increment of shoot length over control was recorded in Zygospora margarita inoculated Cathanthus roseus plant in 180 days harvest, followed by Acalospora levis. And in case of root growth, the length of root in 3 different again M inoculum in Kalanchopunata has increased considerably in all 4 harvesting period, but this was not significant over their control counterpart in all cases. The highest growth in root length in Kalanchopunata was recorded in Acalospora levis inoculated plant at a 180 days harvest, followed by Glomas mossi. And Cathanthus roseus again show, you can show, I will show later you, that Acalospora levis actually increased its root length also, and these are significant. And this is the result in tabular form. You can show it in 120 days treatment. Here I am showing you on Cathanthus roseus only, not Kalanchopunata. Here you can see that all the parameters, that is leaf area, mean leaf number, shoot length, root length, dry biomass, wet biomass of the whole plant showed better result against uninoculated control one for all type of M fungal inoculation. So you can say that M biofertilizer is better than uninoculated one. That is, M biofertilizer is working on these host plants. And this is graphical explanation simply. And here you can see that this is the control one. First one is control, okay, and then last 3 are the inoculated plants. And interestingly, you can see that in case of root length, Acalospora levis increasing root length much more against the other 2. And here you can observe all types of inoculum do not maintaining same type of effect. That is, Acalospora is good for root length increase, Zygospora is better for shoot length, leaf number, leaf area increase. Hence, it is necessary to work on the proper biofertilizer application for proper crop production and meat. Again, this one is graphical explanation on yield, and you can see all the M inoculation show better result against control, and Zygospora margarita again show you the better result. Now, green matter and dry matter production. I am using this result. Okay. In this slide, I am trying to compare the result of two different plants, and this one is Kalanchokinata and this one is on Cathanthus roseus. You can see that during the 90 days period, 120, 150, 180, all the cases above ground biomass is increases, but the rate of increase is more in case of Acalospora levis. Here also, in case of Acalospora levis, but you can see after the fifth month, it is decreasing in case of Acalospora levis, but in case of Zygospora margarita, it is increasing. So these are to be observed very acutely before the production of any biofertilizer or apply on any crop. Again, I am trying to compare the root volume. Here again, you can see that increase of root volume in case of Acalospora levis is quite good, but in case of glomus mossi is not good. But here also same result is seen. Now the biochemical part. We can observe the morphological part in our naked eye and we can compare. But what is within the plant's leaf, shoot, and their biochemical parts, contents. So, Haminoculation effect on chlorophyll content on two plants are observed depicted here. Total chlorophyll content in the leaf tissue of Kalanchokinata haminucleated plants showed here and Cathanthus roseus showed here. And you can see over the control, this one is a control, this one is control. In every case, they are increasing. Highest amount of chlorophyll was observed in glomus mossi inoculated plant at 180 days harvest and 150 days harvest of same inoculated plant. Least amount of chlorophyll increase was observed in Zygospora margarita. But in previous case, you observed that Zygospora margarita is very helpful for the increase of plant total biomass and other parameters. But in case of chlorophyll content, it is not so good. And in case of Cathanthus roseus, you can see this result. And this one is Zygospora margarita. This is decreasing. Okay. Next slide. Now I am trying to show you here the carbohydrate content. Carbohydrate content in the leaf tissue of haminucleated Kalanchokinata showed increase in carbohydrate content over control. And in statistically, though these are not significant, but increasing. But in case of Cathanthus roseus, they are statistically significant and they are increasing. In all the cases with the days, increasing of days. Now the protein content, again, this one is increasing with the application of inocula with the date, increasing date, and everywhere, though they may not be significant, but they are increasing. So next come mycorrhizal dependency. Why mycorrhizal dependency? Actually, more than 90% angiospermic plants are mycorrhizal associated with mycorrhizae. They are dependent actually on mycorrhizae. And to know their mycorrhizal dependency, Jardiman first coined the term, and the mycorrhizal dependency is the degree to which a plant species is depend on the mycorrhizal condition to produce its maximum growth or yield of a given level of soil fertility. So how we can calculate mycorrhizal dependency? First, we have to take the dry weight of mycorrhizal plant and dry weight of non-mycorrhizal plant. We have to subtract this. from dry weight of mycorrhizal minus dry weight of non-mycorrhizal by dry weight of mycorrhizal plant into 100, and this is the calculation of mycorrhizal dependency. Here in this slide you can see that mycorrhizal dependency of these two experimental plants, that is Kalanchoe pinnata and Catharanthus roseus. Now you can see that Catharanthus is more dependent on mycorrhizae for all the time period of their growth and experimental period actually, but and significant for all the applied AM biofertilizer. But in case of Kalanchoe pinnata, this is not so. So when we want to apply biofertilizer, actually Kalanchoe need it more or not, or Catharanthus need it more or not. This is the question. So this is the this should be one objective of research: which plant is more dependent on mycorrhizae, and would we take these mycorrhizae as biofertilizer for long run, or it may be effective or not? Okay. Here, Kalanchoe pinnata after 180 days of harvest in pot culture. After uprooting, you can see these three. For mean calculation, we have to need 3 plants. So here are 3 plants. These are the control. These are the control against the other inoculated plants. Here the Catharanthus roseus, and actually you can see very easily how the difference of their growth. After uprooting and clearing the roots, you can see. After finishing this, okay. Now I come to role of ectomycorrhizae in forestry. Actually, most ECM plants, who are strictly ECM plants, they are not dependent on M fungi. So I divide this my topic into, and a role of ectomycorrhizae in forestry is here. ECM directly involved in the mobilization, absorption and translocation of soil nutrients and water to the host tree roots. ECM actually increase the inorganic phosphate transfer and the increase of inorganic phosphate transporter in the fungal and soil interface. So you can see here how they are connected with the long ECM plants, and these are the fungi. And actually you can see the food bodies. Most of the plants are basidiomycota, under basidiomycota. In forest ecosystem, ectomycorrhizal and saprophytic fungi play a central role in the breakdown of soil organic matter. You can see here. So competition between these two fungal guilds has long been hypothesized to lead to suppression of decomposition rates, and this is known as Gedgill effect. And ECM play an important role in seedling survival. Establishment and growth. Since 1950s, mycorrhizal biofertilizers are used on seedlings in plant nurseries. They improve nitrogen absorption mechanism of the host by emitting protease, chitinase and other enzymes. ECM colonizing the host roots, providing protection of the host against soil pathogens. Again, ECM fungal mycelia connects different species of plants, which reduce competition among plants and contribute to the stability and diversity of the ecosystem. Hence, ECM fungi are widely used in forest management and restoration of ecosystem. In the restoration of ecosystem, the biofertilization, the bioremediation, and the biocontrol of soil, pathogens are the prominent roles of the ectomycorrhizal fungi. These are the some ECM, and these are the food bodies. You can observe easily in the pine forest or some are in our sal forest also. They are present like Russula delicata. But I want to emphasize on M biofertilizer production. During my research period, uh, actually pure culture is very hard because of contamination, but there are so many steps you can take to prepare biofertilizer. Uh, from the Center for Natural Biological Resources and Community, that is CNBRCT Bangalore, for mass culture, I bought uh, pure culture, that is Glomus mossi, Acalospora levis, and Jagaspora margarita, and the three are in packet form. I will show you later slide, next slide. And after collecting these pure fungal spores, fungal spores, first I have to make the media to make the mass culture. And previously I said that they need a simple media, that is sand soil mixture. And to make free the sand soil mixture without other microbial intervention, first of all I have to autoclave it for 1 hour at 121 degrees Celsius and 15-pound pressure, not for single day, for consecutive 3 days. Then sterilized earthen pots filled with 2 kg sterilized sand soil mixture were taken for mass culture, and 20-gram pure inoculum, which was bought from CNBRCD, was used. As a host, sterilized Sudan grass seeds were used. And filter water was sprayed as and when required for 3 months to raise the seedlings, as well as to infect rootlets and production of M spores in pot soils. And these are the packets from CNBRCD. After culture, this on the Sudan grass, you can see. After culture, you can see this type of infection under microscope. Okay, infection threads, and you have to know actually the fungal infection has been made or not, the interaction is made or not. So first of all you have to uproot the plant after 1 month to see the interaction under microscope, and then you have to make the wait, no, not to make, you have to wait for 3 months for the mass culture. And these are the spores in soil medium, glomerular spores, and these are spores. And these are the structures: arbuscular vesicles, acalospora. Acalospora vesicles are like this, and these are the arbuscule, and these are the vesicles within the root cortical cells. And how to harvest you? After 3 months, final production of inoculum, harvest was made. Then store this inoculum or mass-produced inoculum in a cool place away from direct sunlight and heat. And dried soils with spores, including dry roots of Sudan grass and remnant hyphae, may be packaged in a sterilized polythene container at room temperature up to 2 years, and they may remain viable for further use as biofertilizer. And these are the state-wise biofertilizer production. It is data collected, and this is data from 2013 and 14 years 14. And here you can see that Madhya Pradesh using M most, followed by Uttarakhand and Karnataka. And next, this is a state-wise carrier-based biofertilizer production, and Tamil Nadu is the most biofertilizer producing state according to report. And They are producing 22%. And next, you can see that if we divide them in zone wise, then south zone is more biofertilizer producing zone, and within India they are about 47% producer. So uh, last, I am coming to the conclusion. So biofertilizers, being essential component of organic farming, play vital role in maintaining long-term soil fertility and sustainability by fixing atmospheric dinitrogen, mobilizing fixed macro and micronutrients, or convert insoluble phosphate in the soil into available form, thereby increases host plant efficiency. Next, biofertilizers are environment friendly and alternative to chemical fertilizers and have gained prominence in agriculture and forest nursery, though their action is slow and have limited shelf life as compared to chemical fertilizers. Among several different microbial genera and consortium, most popular biofertilizer producing microorganisms are fungi. They decrease the occurrence of plant disease by inhibiting pathogenic microorganisms and suppressing a number of biological processes of the pathogens. Fungal biofertilizer produces hormones and enzymes which stimulate plant growth. Many ectomycorrhizae are potential wild edible mushrooms, as I see you in slide. These fungi play a vital role to establish seedlings in natural ground and widely used to produce ectomycorrhizal forest seedlings in nursery bed artificially for reforestation purposes. In context of both the cost and environmental impact of chemical fertilizers, excessive reliance on the chemical fertilizers is not a viable strategy in long run because of the cost, both in domestic resources and foreign exchange involved in setting up of fertilizer plants and sustaining the production. As VAM or AM is easy to handle and need no synthetic media for mass production, it may be incorporated as biofertilizer in present day scenario. Mycorrhizae are symbiotic and non-pathogenic, hence their potential and applicable for all types of plant growth and fungal biofertilizers. And here are some photographs of now using or applicable biofertilizers and easily available in market. Like now this one is not available and these are available, and only this one is myco apply, this one is ectomycorrhizal, and all others are M fungal biofertilizer. And they can be used they can be used twice in a year, before the onset of vegetative stage and before the flowering also. These are available in Amazon also. So thanks to all the organizing committee members and managing members, managing trustee of AIHSHEW, Honorable Chancellor Sir, Honorable Vice Chancellor Madam, Registrar Madam, Dean of Social Sciences, Coordinator Madam, UGC Study Project, and convener and organizing secretaries, including all the faculty members of Department of Botany, Zoology, Biochemistry, Biotechnology, and Bioproperties, and physician assistant, Avinashilingam Institute for Home Science and Higher Education for Women, Coimbatore, Tamil Nadu, for giving me opportunity, and thank you to all.
Thank you, ma'am, for the excellent speech on biofertilizers. It was a vast study on fungi.
Thank you very much, ma'am, for your unwavering attention throughout this presentation. You have filled our brain with more ideas through this lecture. Thank you, ma'am. Now the forum is open for discussion.
There is a question in the chat box. Amri ma'am,
Okay.
what is the difference between biostimulants and biofertilizer? As European countries prefer to say biostimulants over biofertilizer.
Actually, biostimulant, they are the products that reduce the need for fertilizers and increase the plant growth resistant to water and biotic stresses. And the example as humic acid, fulvic acid. Actually, they comes from several microorganisms. Okay.
Yes, ma'am. Yes, ma'am. Hope the participant got the answer. There is one more question from Mr. Praveen Roylavar. I am surprised why endophyte like Piriformospora indica is mentioned as a biofertilizer.
Uh, Actually, I am working on BAM and I want to stage on M mycorrhizae by fertilizer. That's why I forget to include it, include it. Sorry for this. Next time I will include it. Actually, dark sected and mycorrhizae.
Yes, ma'am. Thank you. Thank you, ma'am. Participants, any other question?
Ma'am? Yes, ma'am. Ma'am, Catranthus roses is very much used for cancer properties. Did you see any changes in the flower content or any increase in sizes while treating with BAM inoculum?
Actually, I have done my work for only 6 months, and during that time, no flowering has been taken. And due to lack of any instrument, I was unable to calculate the alkaloid content of the leaves.
Thank you. Okay, ma'am.
And if you see, because actually I want to quantify the alkaloid, but it was not possible for me. But after viewing the color of the content of by using chemical, it is changing, and it is darker than the control one. That's why I can say that alkaloid content also increases with the fertilizer application.
Okay, ma'am. Thank
Ma'am,
you.
can I ask a question?
Yes, madam, sir.
Dr. Pradeep Desai from Ahmedabad. You had shown in one of your slides at the end of the presentation VAM products, VAM. Are these micronutrients or biofertilizer? Or?
These are actually produced from the not laboratory product. They are producing from the company, and I am not using all this. I am taking it from net, and these are actually they are written as somewhere written as biofertilizer, somewhere written as VAM biofertilizer. Actually, what content are there? I don't know.
Okay. Thank you.
Sir, if you want to know anything about this biofertilizer, CNBRCD from Baghera Sir's laboratory, you can know about more information. You can get more information from this Bangalore, CNBRCD Bangalore. They are producing pure culture for experimental purposes, sir.
Okay, thank you, ma'am. Thank you.
Any more question? So,
If there is no question, shall we move to lunch break, ma'am?
Yes, ma'am.
Now we'll be having a short break. After lunch, we'll be joining in the meet on 11:15, 1:15, sharply at 1:15.
So thank you to all for listening.
Thank you, ma'am.
Thank you very much, ma'am.
Thank you.
Good afternoon, sir.
Good afternoon to you.
Welcome, sir.
Yeah, thank you. Yeah,
Can we start, sir?
yeah, sure. Yeah,
For
yeah.
5 minutes.
I'm ready.
Only minimum participants are there.
Yeah, wonderful. Yes, we can start.
Okay. Thank you, sir.
I think, ma'am, we will wait for 2 to 3 minutes.
Oh, okay, ma'am.
Only 32 participants are there. Good afternoon, sir.
Good afternoon.
Good afternoon, sir.
Oh, sir, madam, good morning. Good afternoon.
லஞ்ச் முடிச்சிட்டீங்களா?
Oh, yes.
சரிங்க. காயத்ரி,
மேம் சொல்லுங்க.
ஸ்டார்ட் பண்ணலாமா?
ஆமா, பார்ட்டிசிபென்ட்ஸ் மினிமமா இருக்காங்க. ஒரு 5 minutes வெயிட் பண்ணலாமா?
வந்துருவாங்க.
பண்ணிக்கலாமா? கண்டனம் பண்ணிக்கலாமா?
என்னப்பா?
பண்ணிக்கலாமா?
ஸ்டார்ட் பண்ணிட்டா வந்துருவாங்க. வந்துனே இருப்பாங்க.
ஓகே, ஓகேங்க மேம்.
Okay, Raji, you can start, Raji.
Raji? You can start the session, ma'am.
Yes, ma'am.
ஸ்டார்ட் பண்ணிக்கலாம் காயத்ரி.
Yes, ma'am.
ஸ்டார்ட் பண்ணிக்கலாம். ஜாயின் பண்ணிடுவாங்க அவங்க.
Okay.
சார். சார் ரெடியா இருக்கார். ஸ்கூல் ஸ்டார்ட்.
Raji, you start, Raji.
Yes, ma'am. Okay, ma'am. Good afternoon, one and all present here. Now we'll begin the technical session by introducing the chief guest, Dr. B. Daniel. Currently, he is Director, Research and Education, Zoo Outreach Organization in Coimbatore, in Chamber Commission, SSC. He served as member of South Asian Invertebrate Specialist Group, member of Invertebrate Conservation Subcommittee, and member of Crocodile Specialist Group, CSC Education. Since 2009, he serves as Associate Director, Journal of Threatened Taxa. 2010, Associate Director, LIPSI, Journal of Tropical Entomology. 2014, Technical Advisor, Life Trust. He is also a professional member in International Zoology Educator Association, Zoological Society of London, life member of BRINC. He also conducted 120 conservation education workshops in India and South Asian countries, and he has also traveled 37 countries on occasions related to wildlife conservation activities and published 78 articles, 5 books, and 4 chapters. Over to you, sir.
Thank you so much for the wonderful introduction. First of all, I would like to just thank the management of Avinashilingam Institute for Home Science and Higher Education for Women, the head of the Department of Zoology, and all the examples from what we have from our studies. So this global warming has been linked to a decline in the population numbers of many species in terrestrial, freshwater, and marine ecosystem as well. So so why are some species are more prone to climate change, while some species can tolerate? We can't just say every species will disappear. Some species can adapt themselves to the changing environment, but certain species which are very sensitive to the environmental situation, maybe based on life history, maybe based on the ecological system that they have, or the behavioral systems, or the physiological condition of the species, or the genetic trait, they may be prone to climate change on a higher level. So that's what we are going to just see now. So some of the predicted aspects of climate change, the effects that those are likely to have on species are, one is phenology. Phenology is a study of cyclic and seasonal natural phenomena, especially in relation to climate and plant and animal life. So that's phenology. We know that season, arrival of the season, right? We say summer, winter, right? We also say that, the summer has just arrived 2 weeks early. Sometimes you just feel that, in January 14th itself, in southern India, the summer will just set. It happened some 4 years ago. That's early setting. That is a seasonal arrival, early setting. So growing seasonal. In certain other cases, the winter or the rainy season may have a longer duration. That's again a problem. It will be a problem for the species. You cannot just, that may have some imbalance with the behavior of the animals, or with the rhythm of the plants that we have in our region. So phenology is something we need to be just, pay some attention. Temperature, of course, everybody knows. Mean temperature, extreme temperature, variability among the temperature, and seasonality, right? So this, the temperature is varying, temperature is increasing. This is one of the predicted aspects of the climate change. The next one is rainfall. Of course, mean rainfall. There is a huge amount of rainfall over a short period of time. That's, we call it as extreme condition of the rainfall. And variability, fluctuation in the rainfall. In certain years it will rain, in certain others it won't rain. Seasonality also. The season won't set, the rainfall won't come. In Kerala, we just say that on June 1st, the rain should come. But no, it so happened that, probably, let's wait and see this year. Probably it may be exceed, maybe post, maybe on a later period, it may appear maybe a week, or even just a week before or after the suitable period. So this is, we call change in the rainfall seasonality. And the extreme events, we are already facing it. Floods happened very recently in Kerala, very often happens in Orissa and certain other parts of the coastal sides. Droughts, it has happened in 70s, we had huge droughts, right? Storms, very frequently we are just getting storms. Of course, in certain cases it's natural, but the current, if we, on a global scale, if you just get some data from the US and certain other Southeast Asian countries, you can just see the frequency of the storm as just, is just very, very high, just increasing. And then the next predicted aspect of climate change is the carbon dioxide concentration. This is where we are just more concerned about it. The atmosphere should have a specific concentration of carbon dioxide and various other gases. We call it as greenhouse gases. If there is an increase in any of these gases, or in the percentage of these gases, probably that, not probably, it will lead to just complications, right? And also the carbon dioxide concentration in ocean also, it matters a lot. We'll talk about it in a few minutes time. So how they affect the species and the ecosystem and various, let's see that. With regard to the phenology, if there is a late setting of the season arrival, or early setting of the season arrival, or if there is a growing length of the season, or shortening period of the season, then it will lead to desynchronization of migration or dispersal events. We all know that, animals, especially with special reference to birds, they migrate longer distance. Just imagine, birds from the American continent, they have to just migrate to the to the southern part of the globe. where it will just look for a warmer region. But when they come here, if it is not warm and if there is no water there, just imagine what will happen. So if there is a late setting of the season, probably that is what we call desynchronization of the migration or dispersal events. So the second thing that could happen is uncoupling of mutualism. Example is just pollination, pollinator laws, right? We all know that pollination is mostly done by, of course, not specifically, mostly majority of the species are done by, majority of the pollination is done by insects. I will give you one example from orchids. Certain orchid species require special insects for the pollination. If there is any delay in the phenology of season setting, or there is a longer duration later, or what will happen, the orchid blooming will just be delayed, and there will be a huge uncoupling of the mutualism, that the pollinators cannot go and pollinate things, and then that will be a lot of problems with the species itself. This is one example. We can give plenty of examples of something. Next thing that could happen is uncoupling of prey-predator relationship. I'm going to give one good example later on. How, if there is a delay in season in phenology, if there is a change in phenology, how that will affect prey-predator relationships. Okay, so the next one that we just think of is the parasite-host relations. Again, I'll give you an example. The interaction with the new pathogen invasives are also related with the phenology. New pathogens could have come, new invasive species could occupy a region because of the delay in the phenology, change in the phenology. So these are some of the examples. So if you, let me just say a few things at this point. If you have any questions, please note it. And if you have any questions, if you can just type it in the question box, you're most welcome. I'll be more than happy to just answer at the end of it. Okay. So this is the second predicted aspect of climate change is temperature. You can just see a range of things that, apart from whatever for all first information that I told you, there are also other changes that would happen. Could be the changes in distribution range. A lot of examples can be given. We can give an example of work done by the Department of Zoology under Mani Meghale, where they did some work on mosquito distribution range. They could also just predict if there is a new introduction of the species in certain regions. So that kind of work can also be just taken up. So this forum is just to introduce certain area where we can just take up new areas where we can just take up some research work also. So maybe we can discuss that part also. So because of temperature, loss of habitat, increased physiological stress, right, changes in fecundity leading to change in population structures. I'm going to give you a good example. Changes in sex ratio, just because of the temperature in the atmosphere, it could happen. Changes in competitive ability. So that could also happen, right? So there are other things with regard to the rainfall, predicted aspects of climate change. So all these things, whichever I just mentioned, could happen. So the other predicted aspect is extreme event. Again, because of this extreme event, all those 4 points which I've marked could happen. The changes in distribution range, habitat loss, increase in physiological stress, changes in fecundity happen. So the last one is carbon dioxide, right? Carbon dioxide. Because of change in the carbon dioxide concentration in the atmosphere, as well as in the ocean and ocean water, all these new things could happen. The one thing that I want to mention is inability to form calcareous structures and dissolving of argonite. So these are something. This is related with calcium setting in corals. Corals, you all know that they are very slow growing. It will take years, maybe decades or centuries for a growth of the setting of the corals. And then the specific concentration of carbon dioxide in the water is just necessary, and the temperature should also be very apt for the calcareous, the deposition of argonite on the coelenterate. Okay, if there is any difference in the carbon dioxide content, what will happen? The sea water will just become acidic. That will lead to dissolve the particular argonite content, the calcium calcareous structures. That will harm the entire ecology of the coral ecosystem itself. So that's we are just concerned about it. Now, How do we know which species are most at risk? I'm just going to give you 5 points, 5 groups of traits that make species susceptible to climate change. One is specialized habitat. You can think of so many species of plants as well as animals. For in case of orchids, certain orchids require specialized habitat. In case of certain microbes, it required specialized fungi, they required specialized habitats. In certain case of animals, say for without example, I can give you hermit crab, where certain crabs just go and inhabit the mollusk cells. They are called specific habitats. Okay, so without it, they cannot just survive. Okay, so species that has specialized habitat or microhabitat requirements will be just prone to climate change problems, right? The second point is narrow environmental tolerance or thresholds that are likely to be exceeded due to climate change at any stage in the life cycle. Narrow environmental tolerance. I can just give examples of amphibians. Amphibians are very, very sensitive to temperature. And if there is any change in the temperature, probably that will lead to the death of the animal itself. We can all just say a few examples from fish. Certain fish like salmon, which is economically important. When they migrate to the fresh water to lay its egg, the temperature should be specific. If there is a slight change in the temperature, it cannot breed. Even if they lay the eggs in the water, fresh water, it cannot just hatch. So a narrow environmental, so that means these species have very narrow environmental tolerance thresholds, which could be altered because of the climate change, and then that will lead to some complications with the species survival, right? The third point could be dependence on specific environmental triggers. I can just give you one good example where the tadpole, if you just go to the high elevation in the hills, you can just say tadpole will be just like a fish, right? Because for want of iodine, these tadpoles won't metamorphose into adults. I can also just some triggers. For example, I can give you one more example. Nothing but these termites. All in a sudden, after a sudden rain, you can just see plenty of termites, flying in a nuptial flight. We call nuptial flights, and then just fly and just fall in front of your tube light or light itself, right? So that's because how they come out immediately, because when there is a rain, after a long period, when there is a rain, the rain brings a component. When they fall on the water, they brings a component called cyanocobalamin. So cyanocobalamin just required for that particular insect, termite, to just transform into immediately winged insect when they just fly. Okay, so these are some of the specific environmental triggers. Okay, so if there is any change in such kind of triggers, imagine if there is no rain for 5 years, what will happen? Tadpoles will remain as tadpoles for a longer time, and the termites will be like that for a longer time, right? So the fourth point is dependence on interspecific interactions that are likely to be disrupted by climate change. I'm going to give you one good example of an Arctic fox. You will like it to know about it. And fifth point is poor ability to disperse or colonize a new or more suitable range. Okay, poor ability to disperse in the sense, certain species are good at dispersal. For example, birds. Certain animals are not good at dispersal. We say, for example, mollusks. And certain other insects, they have very poor dispersal ability. Even fish, we can just say, wherever only they can just migrate one to certain locations only or move, right? So they are also prone to climate change, right? For climate change impacts. So I told you I'll be just giving you some examples. We all know about polar bear because polar bear live in the polar region. But I'm going to give you some examples other than polar bear, where just to show that how in certain regions of the world the species are affected due to climate change. So one is Arctic fox under climate change. I just want you to just read this last point. So this example will highlight climate change's disruptive effects on interactions between species. Okay, you all know Arctic fox. They live in the polar regions and they are predators. They prey on Small mammals. Small mammal in the sense like rodents. When I say rodent, it's like, rat and mice. Small rodents, they live in the crevices of the ice where these Arctic fox feed on them, right? Now, imagine there is an increase in climate change. There is a global warming has happened. And then in the temperatures is slightly, gone up by 1 degree C. What will happen? The ice will start melting. That means the small mammal that live in the crevices will lose its habitat. Ultimately, the population of it will disappear. And then the Arctic fox won't get its food. So what it has to do, it has to just to move from the southern region to just slightly to the upper side, or otherwise it won't get food. What is happening in other side? When there is an increase in the temperature, there is another species called red fox. Red fox extend its range to the northern side, and there's a competition between these two species, that is Arctic fox and red fox. Red fox easily drive or just compete with these Arctic foxes, and the population of the Arctic fox could go down. Now you can just see the link between the rodent, the small mammal, the Arctic fox, the red fox, and the climate change. Okay, so this is the interaction. So this example is to highlight climate changes. Climate changes disruptive effect on interaction between species. This is one good example. We have a good article written by Foden, 2008. And then if you are interested, you can just write to me, I will just give you. The well-known example, next example is leatherback turtle and the climate change. And then the point that we want to highlight is this highlight the impacts of rising temperature on sea level rise. Okay, we have a lot of evidence to say that because of increase in the temperature, because of the melting of the ice, the sea water level will just rise, and that will just cover some of the land where many species depend on. Leatherback turtle is just one. What do? They come to the seashore during certain season when they want to lay their eggs, dig and lay hundreds of eggs there. And the eggs depend on the sandy beach temperature, and they hatch into males and females. But what will happen in case of increased temperature? If there is an increase in temperature, all the eggs, that's called environmental influence. We have studied it in ecology. All the eggs will hatch into females. And then there will be only fewer females left in that particular batch. Now imagine what will happen. That's an imbalance in the sex ratio. That is 98% of all that particular batch will become females. Only 2% will be males. And that too, those 2% of males will be hunted or, will be preyed by some other species and nothing will be left. Okay, so that's the problem. So nesting habitat will also be lost because it has already been degraded. The sea level has just gone up and the nesting habitats are just gone. So this is one good example that highlight the impact of rising temperature and sea level rise. The next one is a very, very interesting species that is called koalas and climate change. You must be having this kind of animal as a pouch, maybe a carry bag. You may be having this design like this. It's a beautiful animal. These are marsupials. Marsupial, kangaroo is one example where they carry their young ones in the pouches, right? So marsupials. So what is the highlight of this example? The last point, highlight the effects of climate change to plants and the animals that rely on them, okay? This particular animal rely on a plant called eucalyptus. There are 600 species of eucalyptus in this world. But if we just live in Australia, Southern Australia, and then if you just go there, they depend on about some 6 eucalyptus trees, right? So what do the eucalyptus trees do? They are carbon sinks. All the plants are carbon sinks, right? They absorb carbon dioxide. But when there is more amount of carbon dioxide in the atmosphere, what will happen? The eucalyptus leaves will absorb more carbon dioxide. The carbon content in the leaves will be very, very high. So now the koalas, they feed on the leaves of this eucalyptus, right? When they eat the normal leaf, everything is just all right. It'll be all right. But when they eat the leaves, which more carbon content in there, the digestive problems will start. Okay, it will also lose its natural value. Ultimately, all the young ones that they feed become malnutrition and they will slowly die. what happened actually in Australia at one point of time, ecologists noticed that all in a sudden, in certain forests, all the young ones started just falling from its mother and they died. They were just looking for the reason why it is so. Then they found that the mother are not able to give a nutritious food because the eucalyptus has more content of carbon content in it, and it's not being digested. And the animals are very, very weak because they are not getting its good food because of loose motion and so many things. They are not having good food. Ultimately, when they feed on the young ones, they become malnutrited and then they die also. So that's the reason. You can just see the link between the climate change, plants, and the animals where they rely on the aquatic plants, right? So these are some of the examples. So I just gave you examples. I have a good number of examples, but I just want to see that we need to look from our region, from Indian region, for similar kind of examples that we can just take. Okay, that's what we just really want. This is a good research topic that people can just take up. So every point of time, I'll be just highlighting some research topics that people can take up. So another research point I want to just tell you is that this particular graph. If you just see this graph, the one in yellow is what is the number of species that is being described at this point of time. We all know 19 lakh species are known to science, but scientists say that 30 lakh species are yet to be described. And then the one in green are the species that are yet to be described. If you just see that insects, huge number of insects are yet to be described, right? In case of fungi, when just now, a few minutes ago, we heard some topic about fungi and various other bacteria and things like that. So a lot of species are yet to be described by fungi. And then prokaryotes and different species like nematodes, plenty of species are yet to be described. And arachnids, plenty of spiders, scorpions, centipedes, millipedes. So these are some of the species are yet to be described. So problem is there in front of you. Plenty of problems. Scientists, zoologists, or biologists, conservationists always think that we have named all the species that we have on this earth. But it is not so. There are plenty of species to be described. In the course of time, much before we describe all the undescribed species, it's a very pathetic situation that species are disappearing. Species are disappearing from this ecosystem. We need to just protect them. We need to just give importance. At least let us describe it and find the status of the species so that we will know that this particular species existed and disappeared. We will also get an opportunity to understand what made a particular species to disappear from the ecosystem. That's the point. So I just want to say that the number of species requiring conservation help will increase dramatically as the effects of climate change are understood with greater clarity and focus. So we need to understand. We need to understand what's the problem with species, plants, ecosystem, animal, climate change, atmosphere, and various other climate systems that we have, and then relate one with the other, come up with a lot of examples so that we can understand fully and do some kind of conservation. I am sure at one point of time, species, as IUCN said, more than 37% of amphibians are already under vulnerable status. Many species will require conservation assistance. So we need to just prepare for that. So that's all. That's one part of it. I just finished. So now I just want to just say a few things about climate science. Let's talk about climate change. I'd like to just introduce certain terminology here. So we all know climate change. Climate change is nothing but alteration in the regular weather sequence due to warming up in the Earth's climate system. So this is called climate change. Alteration in the regular weather. So I want you to just just carefully see these wordings. I'll just show you in the next slide. These three words: weather, warming, climate system. Can you differentiate this? What is weather? How do you differentiate weather with that of a climate? And what is climate system? What are the various things involved in it? And how the system influence the warming up of the earth and how the climate change happens. So this is what we are going to see. So this is something which is words, everything very common to everybody, but people just confuse a bit. That's why I'm just concerned about it. If we are very clear with it, then we can just always do it. So what is weather? Weather is the day-to-day condition of the atmosphere in a particular space. So this morning it was cloudy, and then then after around 11 o'clock it was sunny, right? And then it rained also some time ago. Not rain, but drizzling few minutes ago in my area. Now it is cloudy, and then maybe in the evening there will be some light will be there, and then it will just become very cool, right? So this is what we call weather. People used to go to Kodaikanal and then just say that, oh, today's climate is just very good. No, it's not like that. You have to just say today's weather is good. If you really feel that the weather is good and pleasant, then you just say to this weather is good. But let's see what is climate. Climate, of course, yeah. What is climate? Climate is often defined as the average weather. If we just add all the data of the daily weather status and put together, and then make it into a data, and then just and then you just calculate it. That's what we call climate. So climate is described in terms of mean and variability of temperature, right? Rainfall, wind, and various other things over a period of time ranging from months to millions of years, right? Usually 30 years. That's what scientists say that. So now you know the difference between weather and climate. I'll just say once again. So the one is weather is what we experience in each day, right? And then climate is the sum of all weathers over a certain period for a region or for the planet as a whole. We can just also tell it for the entire region. Okay, so that's all about. Now let's understand climate system. So what is climate system? If one wishes to understand, detect and eventually predict the human influence on climate change, one needs to understand the system that determines the climate of the earth and the process that lead to climate change, right? So let us see what are the systems that we have. One is that we have about 5 systems. One is called atmosphere. We all know that, atmosphere. I'll show you some pictures. Second is called hydrosphere. Okay, I'll just tell in Tamil, Vali Mandala. Okay, second is called hydrosphere. Hydro means water, neer kolam. Okay, cryosphere, panic kolam. This is the term I just gave, panic kolam. I don't know what the terminology panic kolam is there or not, but I just coined it by myself. Okay, so the fourth one is just land, biosphere. Biosphere is weird kolam. We all know that, all living things that we have on this. Atmosphere, hydrosphere, cryosphere, land, and biosphere. All these form climate system. Okay, so you can just see here also the picture. Of course, it's highly complicated. Don't worry about it. So don't forget about these 5 major components that form climate system: atmosphere, hydrosphere, cryosphere, land, and biosphere. Right. So I just want to show you what is an atmosphere. Probably you must be knowing, but still I just want to tell you. It has just got many layers. Of course, I have shown only 4 layers here: troposphere and stratosphere and mesosphere and thermosphere. That is also exosphere that goes to 190,000 kilometer distance. Okay, so if you just take the troposphere, it's just where we live. This is a breathable atmosphere. It's only up to, say, for example, 6,000 meters or so. What's the height of the Mount Everest? We all know that 8,800 plus meters is the height of the Mount Everest. So what the people who just travel in Mount Everest just do, they carry oxygen. After a certain level, they can. This is the level that they can just breathe. After that, they cannot breathe. So this level, this is called tropopause, where first level of the troposphere just ends. So if you just see, if you are flying, if you take a plane, probably the pilot will just say that we are flying at the height of 35,000 feet. So this is the height. So you'll be flying this year. So this is stratosphere. The spy flights are the spy plane. We call spy plane. The spy plane can fly up to this level. Okay, so because at this level they can just fly very, very smoothly. So rest of the sphere, layers are this: mesosphere, thermosphere. You just go on and try and understand in your system. So on what basis they are classified? They are based on the temperature, the direction of the temperature gradient. If you just say here the temperature will be roughly around minus 50 degrees C, and then here maybe minus 30 degrees C, and then it goes like that, minus 190 degrees C, and then it just goes like this, slightly lower, and then higher temperature, it'll go to 30 degrees C, right? So on this basis, this atmosphere is just, so we all, everything is just required only on this. So that's all about the 5 climate system. I want to introduce another 3 terminology. In order to understand the climate change, we need to know 3 important, yes, yet widely misunderstood terms. They are nothing but greenhouse gases, right? The second is global warming. Third one is climate change. Okay, so let's see what are these. I know you all know about this, but still I just want to just tell you once again. Greenhouse gases, or the class of gases which can trap heat near Earth's surface. I showed you now this atmosphere, right? It can just trap in this region, especially in this region, troposphere. Okay, all the breathable air is just here. Here is where the all the of course this is the atmospheric, but these are where the Earth atmosphere gases are there, from where all these greenhouse gases will be just stored. The second thing is the greenhouse global warming. Sorry, the second is global warming. So first we spoke about greenhouse gases. All the greenhouse gases present in the atmosphere should have specific percentage. Say, for example, carbon dioxide should be of 70% like that. Nitrous oxide should be, say, for example, 3% like that. Okay, so every greenhouse gas present, there are about some 30 different kinds of greenhouse gases. 95% are 3 different gases. About 95.5, rest of the 0.5% are the rest of the gases. Okay, so greenhouse gases are gases that can trap heat, but they will have a certain specific percentage in the atmosphere. So the second thing is greenhouse gases. As the greenhouse gases increase in the percentage in the atmosphere, the extra heat they trap leads to global warming. Simple. When there is more amount of greenhouse gases in the atmosphere, that will trap more amount of heat, leading to global warming. Okay, so this global warming in turn places pressure on the Earth's climate system. You remember what are the climate system, and lead to climate change. The climate change happens. That will lead to so many other complications. So these are the things. Okay, so some greenhouse gases are carbon dioxide, methane, nitrous oxide, water vapor, ozone. Okay, so what they do, the sunlight falls on the Earth. Certain amount of sunlight is reflected from the atmosphere clouds itself, or the minor particles found there. On certain amount of energy just falling on the Earth and absorbed, it's reflected back, and it's again absorbed by the greenhouse gases found there, and it's just reflected back onto the Earth, so that the Earth will be just maintained at a particular temperature, say 15 degrees C, 16 degrees C on an average. If there is no greenhouse gas present in the atmosphere, then the Earth will go to minus 18 degrees C. That means the Earth will not be suitable for the support of any life forms other than microbes, right? Maybe. So it's very, very important. Greenhouse gas presence is very, very important. Now, the question is, when greenhouse gas is important, why do we blame it? We blame it because of so many other reasons. So this is just a calculation, how much energy just reaches here, how much energy is reflected back, and things like that. You can just read it some other time. Okay, so this picture. If you just see this, you can find a person here. You can call him guardian. You can just see an animal-like thing. This is called nothing but ghost. And this is the atmosphere, right? That's a greenhouse. And this is a biosphere, the animals and all the climate systems representative of the biosphere, of the climate systems that we have, right? I'm just for your understanding, I'm going to tell a small thing, right? That's called 3G. We know about 2G, we know about 4G, now we are getting 5G, but this is nothing but 3G. 3G is nothing but greenhouse gas guardian. GGG, greenhouse gas guardian and greenhouse gas ghost, right? So what I'm just trying to tell you is that a guardian which is necessary for the healthy living of all living things on this earth, Guardian denotes the greenhouse gases, will turn into a ghost if the concentration of the gas increases, right? So that's what we are trying to tell you now. Okay, both are 3Gs only. Greenhouse gases, it will become greenhouse ghost. Okay, it may be slightly difficult for you to understand, but let me give you with another example. We all eat sugar, right? Sugar is very, very important for our daily energy. We take sugar, and that gives energy. Okay, so every day we say, for example, we take 200 or 100 grams or 100 grams. No, that's too much. So say 50 grams of sugar every day, or in some other form, we get a lot of calories, and then we utilize it. Right now, imagine you take a lot of sugar every day. You take 1 kilo of sugar, right? And then what will happen to your health? There will be a huge amount of sugar content in your body. That will just the sugar, which has to be on a specific amount in your blood, will increase. That will be a problem. That will lead to a lot of health issues for you. So the Earth system is exactly like this. The greenhouse gases, which has to be on a specific concentration, when there is an increase in the specific concentration, it will become it will start harming the living things and the entire earth itself, right? So how the concentration of the greenhouse gases increase? It's all because of human activities. We release large amount of greenhouse gases. We release carbon dioxide. We use fossil fuels and petrols and so many things. We release methane. We release a lot of nitrous oxide and water vapor, so many things, and that will lead to concentration, increased concentration of all the greenhouse gases in the atmosphere, resulting in the global warming, leading to climate change. So that's the story I just wanted to tell you. I just want to just show you this graph. This is the amount of carbon dioxide. But until 1700, there was no problem. The carbon dioxide content in the methane content, nitrous oxide content was absolutely all right. After the introduction of the industrial revolution, about some 150 years before, we just started releasing a lot of carbon dioxide, all the other nitrous contents, greenhouse gases, resulting in the climate change system. So what will happen if you continue to do it? Scientists predict that the time is 2:15. That scientists predict that by 2080, the availability of water in the rivers, especially in India, will decrease. Right? The tropical forests and grasslands, I've just mentioned on South America and Africa only. You can also include all the tropical other regions, will die back. Right? The production of the food in India and rest of the continents will decline. Okay, certain other ecosystems like coastal ecosystem, fisheries, aquaculture, freshwater resources, human settlements, and tourism, everything will be affected. There will be huge problem related to waterborne diseases, vector-borne diseases. Heat stress will be there for all the living things. So that this will be, apart from that, all these problems will happen. Hurricane, landslides, insect infestation, flood, drought, and so many things will happen. So that's why we are concerned about it. That's why we need to practice sustainable utilization. In order to just reduce all these things, we need to have a sustainable practice of utilizing all the natural resources. Nilaittanmai, the resources that we have should continue should be there for the future generation also. So I just stated that sustainability means meeting our own needs without compromising the ability of future generations to meet their own needs. Okay, we should also use, the future generation should also use it. So only if we just leave something, they will be in a position to use it. We need to start utilizing all the natural resources sustainably. You know the meaning of sustainability. Okay, sustainability means nilayana payanbadu. Otherwise, valam kundra payanbadu. That's what I meant. Sustainable living means utility means sustainable. Utility means valam kundra payanbadu. So sustainable living describes a lifestyle that attempts to reduce an individual's or society's use of Earth's natural resources and one personal resources also, right? Otherwise, there are so many terms for that also. So we have started utilizing one-time use products, right? I'm using a ink pen. There's an ink pen, right? So at least, I can able to use this ink pen and then just repeatedly use the same material. I don't have to throw my ball pen every time. I don't have any ball pen here every time, because that's a small thing that I can do. And I started using my cycle also. If at all I want to buy some milk, so I'll go to a small shop very, very close by, I use my cycle. I have a bicycle also. So these are small things that we need to just practice and do it. So in 2015, the United Nations adapted the Sustainable Development Goals. It goes about some 17 goals. You can just go and see. I'll just give you the link later. Okay, so you can also go and see all those 17 sustainable goals. And wherever you can just take up any kind of work related to any of fulfilling any of this, maybe you'll be more than happy to. It will be very, very good for the society. And the UN has proposed 6 climate positive actions for governments. It's not just for the government. Government means people. For us, right? Green transition. Investment must accelerate the decarbonization of all aspects of our economy. Less use of fossil fuel. And not only the government, we should also just do it. Green jobs and sustainable and inclusive growth. Green jobs means the work or any kind of industry that will go green. Okay, so that's what we call it as green jobs and sustainable and inclusive growth. Okay, so third one is green economy, making societies and people more resilient through a transition that is fair to and all the levels, no one behind. Okay, so we need to just take every people, include every people, practice all these things. So we need to think of any green economy also. Fourth one is invest in sustainable solutions, right? Let's go for renewable energy instead of using the fossil fuels. Sunlight is there, solar panels can be used for so many other things, and we can also reduce pollutants, right? So the fifth one is confront all climate risk. We need to just learn to confront the climate risk. We cannot just run away from all these problems that we are having. So that means we need to be prepared for it, right? We need to prepare for the next 25 years or 50 years. We don't want all of us to wear huge amount of big size masks around us and go for our daily jobs because the oxygen will not be there in the atmosphere. So we don't want such situation. The sixth one is cooperation. No country can just do things by its own. Okay, every country just cooperate. This is for a global thing. This kind of thing is available with the website. You can just do that. And then I just want to just conclude my talk with this. It's 2:21. So we should learn to live sustainably. Every individual has the responsibility. That is the want. Every individual should be responsible. We cannot just pinpoint somebody else or the government or some other developed country, or developing countries or underdeveloped country. We need to just bring everybody into it. Why I am telling you is that at one point of time, the scholars, the students, you will be the future custodians of this earth, right? And you will be in big positions, right? Whenever you take up big positions in future, when you are given a chance for you to take a decision, keep all these points in mind, and then you need not necessarily be an ecologist, conservationist. Even if you're a police officer, even if you are an administrative officer, you are a forest department, you can always think of it. So the best way is to live more simply. So Gandhi followed the same method of just living more simply. He says that the difference between what we do and what we are capable of doing would suffice the problem, most of the world's problems. So that's what Mahatma Gandhi says. With that, I just conclude my talk. So these are certain things that we can just think of carrying out at individual level, at home, using certain things in a lesser forms. Think of so many other things that we can just help in the reduction of the carbon emission and use of fossil fuels. Okay, thank you very much. I'll be more than happy to just respond to you or any of you in case if you need any kind of help or assistance with regard to species conservation or anything related with climate change, or any research topics and that's required for you, we can always coordinate, right? Okay, I take this opportunity to thank the management, the institution management, all head of the departments, all the faculty members, the students, the coordinators in particular, who are just helping with coordinating the thing. Yesterday somebody helped me a lot to just to do this presentation. So I just appreciate each and every one of you. This is my email ID. I have my official and personal email also. You can also note down my WhatsApp 95977037110. I'll put everything there in the I'll put it in the sorry, am I there? Okay, so I'll put it in the chat box. You can just write to me at any time you feel like doing. So okay, so now I just finished my talk. Just leave the mic to the organizers. Yeah, thank you so much. Yes.
Thank you, sir, for a brief explanation about climate change and environmental sustainability. We are grateful for the rapt attention with which you sat through this lecture. Thank you, sir. Now the forum is open for discussion.
You can also just type in if you have any questions, and I know it's just very broad subject, but if you have any questions, you can write to me also later. And I think there's no question from anybody. Let me check if there is any question in the box. Okay, thank you. Unmute, please.
Hello.
Yes, we can hear.
Hello, sister. Can you hear me?
Yes, we can hear.
Okay. Somebody else is talking. Yeah. Okay.
I think there's no question from anybody. Maybe.
If any questions, you please post it in the chat box. Thank you, sir, for the very informative session. I hope all the participants would have been benefited from your session.
Yeah, thank you so much. Yeah.
Swarna, you can just propose the vote of thanks.
Okay, ma'am. Now, we will begin the technical session by introducing the resource person, Dr. Vidya Lakshmi. She was awarded with Diploma in Reproductive Medicine in Kiel University, Germany. From 2015 till 2018, she worked at
Swarna,
GKNM Hospital. Ma'am?
ma'am is there? Okay. Good
Yes,
afternoon, ma'am.
she's there, ma'am.
Good afternoon, ma'am. I think ma'am is not there, right?
Yes, ma'am.
Let us wait.
Okay.
Let's wait till the resource person joins us, okay?
Okay, ma'am. Okay.
We will let you know.
Can you hear me? Can you hear me here? But I'm not able to hear anybody speaking there.
Yes, ma'am. You're audible, ma'am.
1 second, sister.
Now, we will begin the technical session by
பேசுறது உங்களுக்கு கேக்குது பட் எனக்கு அவங்க பேசுறது கேட்க மாட்டேங்குது இதுல வேணா போட்டுக்கலாமா பரவாயில்ல
Ma'am, do you hear me, ma'am?
Yeah, it's audible, Swarna.
Okay, ma'am. Okay. Now, we will begin the technical session by introducing the resource person, Dr. Vidya Lakshmi, MBBS. She was awarded with Diploma in Reproductive Medicine in Kiel University, Germany. From 2015 till 2018, she worked at GKNM Hospital. From 2018 till 2020, she worked at Sengaliappan Hospital. She currently works as a consultant at the Excel Gynecare Women's Specialty Center. We are fortunate for having you, ma'am. The session is over to you. Ma'am, you are not audible. No, ma'am,
Ma'am, you're not audible, ma'am.
No, ma'am, you are not audible. Not audible.
Ma'am, you're not audible, ma'am. Can you please remove your headset and then plug it on?
Ma'am, ma'am, she's got some technical glitch, ma'am. She'll be again joining in 5 more minutes.
Okay, ma'am.
Okay,
Yes,
ma'am. Okay.
Yes, ma'am. Thank you.
Hello. Hello, can you hear me now?
You're
Hello.
audible now, ma'am.
Audible.
Hello.
You're audible, ma'am.
Hello.
You're audible, ma'am.
You are audible, ma'am.
Can you hear me? Can you hear me now? Hello.
Yes, ma'am, you're
We
audible.
can hear you.
Hello, sister. Can you hear me?
We can hear you.
Yes, ma'am, we can hear you. Please go ahead, ma'am.
Okay. Hello. Hello, can you hear me now?
Yes,
Yes,
ma'am.
ma'am.
Okay, thank you. I think
Headphones
headphones. Okay, no problem. Okay. I think, but they can hear you now. They hear me now.
Okay.
Okay, I'll continue. Check. Uh, Good afternoon to one and all present here. Um, my apologies for the delay. There was some malfunctioning of the speaker system. Um, so I am audible, right? Because I'm not able to hear anything from my side, right?
Yes,
Hello?
yes, ma'am.
I am audible. Okay. So I take immense pleasure in being able to address all of you today. So the next 1 hour, we are going to be talking about cervical cancer, the screening and preventive strategies of cervical cancer. So I chose this topic because it is cervical cancer is not just another health issue. It's not just another disease. It's more of a public health problem, uh, especially in developing countries like India. Can you please go on to the next slide? So what exactly is cervical cancer? In simple terms, it is cancer arising from the cervix. So, to those of you who do not know what a cervix is, this is the image of the female reproductive tract that you're seeing on the screen. So, that is the uterus, which has a cavity inside which the baby grows when someone is pregnant. And the uterus tapers down and forms a small canal down below, which is called the cervix. So the inner part of the cervix that opens into the uterus is the internal os. The outer part that opens into the vagina is called the external os. And the cervix is an organ which is about approximately 3 to 4 centimeter in length and approximately 3 to 4 centimeter in diameter. So a cancer that arises from the cells of the cervix is called a cervical cancer. Kindly move on to the next slide, please. So, the burden of cervical cancer in India. So, this cervical cancer is considered to be the second most common gynecological cancer all around the world. But when it comes to India, this tops the list. And this is the second most common cancer related death that we find among Indian women. And approximately over 5 lakh women die of this cancer every year. And surprisingly, you will not believe at least 25% of the world's cervical cancer related deaths comes from India. And in 2018 alone, we have recorded about 60,078 deaths and 96,922 new cases of cervical cancer. And such is the burden of cervical cancer in India. So it's as much as COVID-19, right? And it's very sad that the awareness among the people of India, the women of India, is still very, very low about cervical cancer. And that is the reason why we are talking about it today. Next slide, please. And the best part of it is that cervical cancer is a preventable disease. It is a 100% preventable disease. Why? Because there are innumerable number of screening methods that are available to detect the cervical cancer even before cancer happens, because the disease goes through a prolonged precancerous stage. We are going to talk about it in detail. And this is the only cancer, one of the few cancers that can be prevented by vaccine. You would have heard of vaccines for infections. That is vaccine for tuberculosis, diphtheria, pertussis. That is vaccine for COVID-19. That is vaccine for innumerable number of infections. But this is one cancer that can be prevented by vaccination. Next slide, please. So what is happening in the developed nations? Why are we telling that the cancer is prevalent highly in developing countries only now? So, most of the higher income countries, the Western world, have successfully reduced the burden of cervical cancer by about 65% in the last 40 years. How did they manage to do this? By frequent screening of women who are sexually active. This helps in early detection of the cancer and removal of cancer in the pre-cancerous stage itself. And as soon as the HPV vaccine was introduced, a lot of countries have adopted this into their vaccination schedule, national immunization schedule, and they are vaccinating the adolescent girls against human papilloma virus 16 and 18, which cause almost 70% of the cervical cancers. Next slide. So what is the situation in India? Situation in India is that screening prevalence in the age 15 to 49 is still only 29.8% according to one of the studies. And lack of a national screening campaign for cervical cancer is a major drawback. And knowledge about cervical cancer prevention and the screening methods that are available is very limited among Indian women. And another major problem is the stigmata associated with the disease. In India, everything has a stigma associated with it. Indian women are very, very shy and they feel very upset to come to a gynecologist and screen themselves for a problem. They don't really take care of their health very well. And especially when there is a problem concerning their genital tract, they are extremely shy to disclose it and even come and approach a doctor for it. That is the problem right now. So next slide, please. So what causes cervical cancer? So we can say the most common cause of cervical cancer is the virus, human papilloma virus. This causes most of the cervical cancers. And there are more than 100 different types of human papilloma virus that have been detected. Okay, the ball-like viral structures that you're seeing on the screen, that is what is human papilloma virus. And at least 14 of them are cancer-causing viruses. And how does it get transmitted from person to person? Every virus, every microbe has its own way of spreading. Like we know COVID-19 spreads through airborne, it's an airborne infection. Same way HPV spreads through sexual contact. And not just sexual contact, even by an intimate skin-to-skin contact, it can transmit from person to person. And among the various subtypes, HPV 16 and 18 causes about 70% of cervical cancers and the pre-cancerous cervical lesions. And what else can they cause? They can also cause cancers in the other parts, the vagina, the vulva, the outer area, that is the outer genital area, penis in men, oropharynx, that is the back passage of the food tract, and anus. Next slide. This human papilloma virus is considered the most common sexually transmitted infection in USA, and at least 40 different types infect the genital areas of men and women. And 80% of women get infected with human papilloma virus at least 1 year after after marriage. Next slide. And fact is that most of the human papilloma virus infections clear within 2 years after the infection, even without any treatment. So how does this happen? As long as somebody is immunocompetent, somebody's immune system is good, they are able to clear up this infection even naturally. And a very small proportion of this infection persists in the genital tract and it progresses to cancer. Next slide. And there are other non-cancer causing types of HPV, that is HPV 6, HPV 11. They cause genital warts, and also warts, you can see warts in the fingers in the picture over there, and respiratory papillomatosis, that is in the respiratory tract. Next slide. It takes approximately 15 to 20 years for this HPV infection to progress to cervical cancer in a person with a normal immune system. So imagine it has a very prolonged course. So from the time a person gets infected, they take at least 15 to 20 years for it to progress to cervical cancer. So it means at any point in this 15 to 20 years, had we screened these women for changes in the cervix through simple tests that are going to cost very less and also less time, we could have detected this cancer in a very, very early stage and saved lives. So that is why it is very important. So what are all the risk factors? I said most of them clear the HPV infection in a span of 2 years when they have a good immune system. So who are the people that are more susceptible to development of cancer? Who are the people in whom HPV does not get cleared? It stays and develops into cancer. So these are the risk factors. It depends upon the HPV type. I told 100 different types are there. So it depends upon the type. When they are infected with the high risk type, that is HPV 16 and 18, which are the cancer causing strains, or when the person is immunosuppressed, who are immunosuppressed, somebody is suffering from some other cancer, chronic diseases, diabetes, chronic alcoholics, smokers, they're all considered immunosuppressed. Patients may be patients who are on immunosuppressive medications. Patients who are on immunosuppressive medications for transplants and other things are more susceptible to this cancer. Co-infection with other sexually transmitted infections like chlamydia, gonorrhea, early age at marriage, like which is the scenario in India, people get married at 17, 18, having multiple sexual partners, parity, that is more number of children. Women who have more number of children are more prone. Early age at first birth, which is again a major risk factor when it comes to Indian scenario. Tobacco smoking. A lot of our rural women still do smoke tobacco. And low socioeconomic status. These are all the various risk factors for HPV to persist and development of cervical cancer. Is more. So how do they come to us? How do cervical cancer patients come to us? They come to us with bleeding in between the menses. Post-coital bleeding after having a relationship, they can have some bleeding. Post-menopausal bleeding, women have reached menopause, stopped bleeding by 50, 60. They come after several years with bleeding problem. So that's when we suspect they can have copious watery discharge, increased vaginal discharge from down below. It can be a very foul smelling discharge that they are very shy. Sometimes they come very late and it can cause lower abdomen pain, pelvic pain, back pain, symptoms like that. So, and once they come to us at this stage, when it presents like this, it is a full-blown cervical cancer most of the time. And we put in a speculum, open up the vagina, we see a big chunk of cauliflower growth or ulcer sitting on the cervix. And it is an established cervical cancer. But when we are talking about prevention, we are talking about a stage when you put a speculum, you don't see anything in the cervix, the patient does not have any symptom, but still they could be having HPV infection and anywhere in the stage that is progressing to cancer if it is left undetected. Next slide, please. So, what is prevention? So, when you talk about prevention of any disease, there are three different types of prevention. One is primary prevention, secondary, and tertiary prevention. So, what is primary prevention? Somebody has risk factors for a disease. Someone is smoking, we know this person is prone to develop lung cancer, he can develop a heart attack, coronary artery disease, he can develop cancers of the respiratory tract, they can develop COPD, a lot of things. But when you catch hold of them, when you say smoking causes cancer, smoking kills. This is primary prevention. You're trying to eliminate the risk factors. You're trying to educate them. This is primary prevention. So what is secondary prevention? Intervention after the disease has started. So the person has risk factors, the disease, the damage has already started. You're trying to detect it at an early stage. That is secondary prevention. And what is tertiary prevention? The disease has begun, but you catch hold of them, educate them about the symptoms, ask them to come forward, diagnose and treat them early. This is tertiary prevention. This is how you avoid the disease from progressing further and developing complications. Next slide. So when it comes to cervical cancer, what is primary prevention? Vaccinating the girls 9 to 14 is a part of primary prevention. Giving health information and warnings about tobacco use, safe sex education tailored to the age and culture that's prevalent in the area or the country, condom promotion, educating the people about the various risk factors, telling them to avoid them, and educating women about vaccinating their girl children, educating the girls. They will go forward and ask for HPV vaccination. This is how we do primary prevention. So, next slide. So, secondary prevention is somebody has already got a HPV infection. Before it progresses and develops into a cervical cancer, we are doing screening by means of small tests like pap smear test, HPV DNA test, and treating the precancerous stage itself. This is called as secondary prevention. And tertiary prevention is when we treat the invasive cancer at any stage, we detect it early and we start treatment before it gets complicated by means of surgery, radiotherapy, or chemotherapy. That's tertiary prevention. So let us talk something about the HPV vaccine. So this human papilloma virus vaccine is a prepared by a recombinant DNA technology. By means of this, virus-like particles are created, and this is what is used for vaccination. And we need to understand that this vaccine is completely prophylactic. That is, it prevents from HPV infection, but it is not therapeutic. It does not treat HPV infection. But somebody, you catch hold of them, children who are not married, young girls before marriage, you give them HPV vaccination, it prevents them against HPV. But women who are already married, who have got HPV infection already, you give them the vaccine, it's not going to cure the HPV infection. And one another important thing is that HPV vaccine, vaccinating a girl is not a replacement for screening. A girl gets vaccinated, then later on she gets married. Still, it is important for her to undergo screening every 3 years or every 5 years, depending upon the age. So there are 3 different types of HPV vaccine in the market. The first one was introduced was Cervarix. Cervarix is a bivalent vaccine which acts against HPV 16 and 18, the 2 high-risk HPV subtypes. And Gardasil is a quadrivalent vaccine. It acts against HPV 6, 11, 16, and 18. So basically, it protects against the high-risk HPV as well as 6 and 11, which are associated with warts, I told you. And Gardasil 9 is a nonavalent vaccine. It it protects against 9 different subtypes of HPV, that is 6, 11, 16, 18, 31, 33, 45, 52, 58, 9 different types which include the high risk and the low risk types. So, Cervarix and Gardasil give 100% protection against the persistent HPV 16 and 18, and Gardasil 9 gives 100% protection against HPV types, including those that are involved in other cancers. Next slide. Next slide, please. Next slide, please. Next slide, please. Ma'am, next slide, please.
Ma'am?
Next slide, next slide. I'm not able to see the next slide.
Just a minute, ma'am.
Oh, sorry.
Ma'am, here also we have some network issue, ma'am.
Okay, all right.
Yes, ma'am.
Ma'am, the slide is visible, ma'am?
Yes, I can see the slide. I can see the first slide.
Yes, ma'am. There are some technical problems, ma'am. That's why.
And that's
I'll
okay.
move on, ma'am.
Yes. Can I proceed?
Yes, ma'am.
Okay. Yes.
Yes, ma'am. Yes, ma'am.
So, what is the recommendation of when it comes to these vaccines? So, who should be vaccinated? You should vaccinate ideally girls starting at the age of 9 to 15. You have to vaccinate, and yeah, I was talking about the efficacy, and the 2019 meta-analysis of girls only vaccination program done in 14 different countries showed that HPV-6 vaccination against HPV-16-18 helped reduce the infection by 83% in girls of age 15 to 19 and about 66% in women aged 20 to 24 years. Next slide. So ideally, you should vaccinate women, girls that are less than 15 years of age. When you vaccinate girls less than 15 years, only 2 doses are given. And if a girl crosses 15 years and we vaccinate her, 3 doses are to be given. The first dose is to be given at least 4 weeks after the second dose is to be given at least 4 weeks after the first one, and the third dose at least 12 weeks after the second one. So who should get vaccinated? Ideally, girls before their marriage, before first sexual exposure, should get vaccinated. But it is not wrong to vaccinate women who are already married also. Whatever protection it offers, let it offer. It is good. Okay. But however, the efficacy is much higher when it is given before the first exposure to the virus. Next slide, please. Next slide. Next slide on side effects. So are there any side effects on taking this vaccination? Yes, there can be pain at the site of vaccine. There can be some amount of redness or swelling like any other intramuscular injection that is given. Very rarely, there have been reports of girls fainting after the injection. So now, what we say is we ask them to sit or lie down for at least 15 minutes after receiving the vaccination. Fever can be a side effect, headache, nausea, muscle, joint pains. These can be some minor side effects that we see with the vaccination. But the protection it offers is worth it. I think most of us experience these kind of side effects even with COVID-19 vaccination. Any vaccination can trigger some amount of fever when it is when it is active. Next slide. Next slide, please. So, a major milestone with vaccine was reached in 2019 when almost 100 countries introduced this HPV vaccine into their national immunization schedule. The next slide. So you can see that there is Russia, Australia, New Zealand, South Africa, almost all countries in the South America and North America have introduced this as a part of their national immunization schedule. You can see the whole of most of Asia, India, Sri Lanka, China, all of this are yet to include this in their vaccination schedule, while we are among the one of the countries that has very high prevalence of this particular disease. So what do you think? I told you women get infected with human papilloma virus after they get married. At least 80% get the infection within 1 year of marriage. So should we vaccinate men? Yes, I think we should vaccinate men because it is not a infection that is common only to women. And to men also, it can offer protection against genital warts, penile oropharyngeal cancer, and most importantly, it protects their partners from getting the HPV infection. And USA and Australia have successfully implemented vaccination of the male children also. Next slide. Next slide, please. So that is about vaccination. So let me tell you what exactly happens in the cervix. So you can see the lower part of the uterus is the cervix. It has got a lining that lines the inner part, that is the endocervix, and the outer part that is facing the vagina is called the ectocervix, as marked in the diagram. This endocervix is lined by block-like cells, which is called the columnar cells, and the ectocervix, which is open into the vagina, is lined by squamous cells, stratified squamous cells. And the junction where the endocervix and the ectocervix meet is called the squamocolumnar junction. So what happens is that when a girl grows up, when they get exposed to the endogenous hormones, the estrogen, a part of the ectocervix gets exposed to the vagina. While only the ectocervix, which is lined by the squamous epithelium, is adapted to managing the vaginal pH, while the endocervix, which is lined by squamous epithelium, does not act as well as squamous cells in adapting to the vaginal pH. So what happens is that when the cervix opens up and the columnar cells get exposed to the vagina, it undergoes constant irritation. So in response to the irritation, it adapts to some amount of changes, undergoes changes in its own anatomy, in its own histology, and tries to become squamous cell in order to adapt to the new environment. And this process is called as metaplasia. But however, the columnar cells that have transformed into squamous cells are not natural squamous cells. They can't act as well as natural squamous cells. They are just metaplastic squamous cells. And ultimately, there is a new squamocolumnar junction that is formed. Next slide, please. So as you can see over here, the original squamocolumnar junction is outside, lined by the dotted lines, and a new squamocolumnar junction has formed inside with a reddish area, just outside the reddish area. So the area that is in between the original squamocolumnar junction and the new squamocolumnar junction is basically not the natural squamous cells, but it is the endocervical cells that has become squamous cells. That is the metaplastic squamous cells, and this area is called the transformation zone, and this is the zone that is more prone for developing cancer. Next slide, please. So, as I told you, once the HPV infection occurs, before it becomes a cervical cancer, as it persists, it goes through a lot of changes in the cervix before becoming cervical cancer. That is, it goes through a stage of low-grade squamous intraepithelial lesion, LSIL, and it becomes HSIL, that is high-grade squamous intraepithelial lesion, and then it progresses and persists and over a period becomes cancer of the cervix. And when we take a biopsy, we through colposcopy, we say it goes through CIN1, CIN2, and CIN3, that is cervical intraepithelial lesion 1, 2, and 3. It goes through 3 different stages of progression before it becomes cancer of the cervix. Next slide, please. So you can see in this picture, the first one is the histology of normal cervix. So here you can see that the cells are all very pink, rich in glycogen. You can see that the cytoplasm is more, the white portion is more, the pink portion is more, while the nucleus is seen as very fine dots. The nuclear cytoplasmic ratio here is maintained in a normal cervix. And as it progresses to from CIN1, 2, and 3, you can see that in CIN1, the lower 1/3 of the cells are showing some amount of changes. The changes basically is the nucleus becomes larger and the cytoplasm becomes smaller. There is alteration of the nuclear cytoplasmic ratio. There is hyperchromasia. The cells are looking more darker and dividing more rapidly. And as it progresses to CIN2, you can see that almost 2/3 of the cells, 2/3 layers is being occupied by these kind of cells, and the nucleus is becoming larger and larger and more hyperchromatic. And in CIN3, almost the entire layer is completely involved by the dysplastic cells. So this concept we are going to use in screening. I will tell you how. So when it comes to the Western world, each country has its own national screening protocols. So in the US, as a woman reaches 24 and a half years of age, every woman that reaches that age receives a letter from the NHS that you are about to reach 25 and you have to enter the cervical screening program here onwards. And every 3 years, she gets a letter inviting her to come and get the cervical screening done. And this happens until they reach 65 years of age, and by 65 they will be cleared and discharged from the screening program, provided their last 3 smears have been normal and they have told you're all clear, no problem, no chances of developing CA cervix. So by 65 years, provided last 3 smears are normal, they discharge them from the screening program. Same way, just like in the UK, US also has its own screening program, and many of the Western world have adopted a screening program suitable to their system. So, according to the NHS screening program, from 25 to 50 years of age, every woman undergoes a cervical screening every 3 years. And starting from 51 to 65 years, they have to undergo cervical screening every 5 years. And so, what are the various modalities of screening? So, we can do a clinical downstaging via villi, cervical cytology, which is the most popular method of screening we have been doing, HPV DNA testing, and colposcopy. Let's see what they are. Next slide, please. So what is clinical downstaging? Clinical downstaging is basically when a woman walks into the clinic with symptoms. I'm having bleeding, I'm having a very bad smelling discharge. We put in a speculum, open up the vagina and see whether the cervix is showing any changes, ulceration, growth. And we take up, then we diagnose early stage cancer. This is what is clinical downstaging. And if they have any abnormal findings, it needs to be further investigated. And this method is not at all intended for detection of the disease at a pre-cancerous stage. This method of screening is basically a tertiary prevention strategy that can be done once a person develops a cancer. You're trying to detect it early when they come with symptoms. And this method can only be recommended in a very low resource setting, where pathologists are not available, doctors are not available, staff nurses who know to do a pap smear are not available. In that kind of a setting is where you can do this clinical downstaging as a modality of screening. But it's basically not a screening method at all. It's just for early diagnosis. Next slide. Visual inspection under acetic acid, visual inspection under Lugol's iodine has been popularly used in many areas. Again, in areas where manpower resources are less, where you do not have many laboratories to read the smear, or you do not have trained staffs to prepare the smear and read it under a microscope and give a report. So this is used for large scale. This has been used for several years in large scale rural camps and all those things. So what is the concept of this? Via villi. Next slide. So as I told you, there is this squamous epithelium which is native to the ectocervix, which is a normal epithelium, and then the endocervix comes out. It tries to change, adapt, and form a metaplastic squamous epithelium, which is more prone for developing this pre-invasive cancers and cervical cancers. This squamous epithelium, like I told you, is rich in cytoplasm, and cytoplasm is rich in glycogen, and it has very less nuclear protein. Nucleus is rich in protein, and cytoplasm is rich in glycogen. This is the concept. While in a metaplastic epithelium, there is a poor glycogen. Cytoplasm is very less. Nucleus is very large. There is a lot of protein. So what happens? Next slide. When you put acetic acid on the cervix, what happens? Any acid that coagulates the proteins, and protein coagulates and becomes white. So this is the concept. You put acetic acid to the cervix and just look at the cervix. It becomes white. It means there is metaplastic squamous epithelium. If it is a native squamous epithelium, which is normal, it's rich in glycogen, no coagulation, it remains pink. So what happens when you put iodine? Lugol's iodine is what we use, and after acetic acid testing, we put iodine over the cervix. We smear iodine using swab stick all around the cervix, and we look for changes in the cervix. So what happens? Iodine acts on the glycogen and turns it mahogany brown. So what turns brown in color? A normal squamous epithelium turns brown in color, mahogany brown in color. But if it is the CIN that is sitting over there in the cervix, what happens? There is no cytoplasm, it's just full of nucleus, protein material. It does not turn brown, it remains yellow. This is a simple color coding concept that any person you teach to a staff who is not trained, any newcomer, a trainee, they will understand. You put acetic acid, it turns white, it is abnormal cervix. You put iodine, it turns yellow, it is abnormal. Remains yellow, it is abnormal cervix. So this has been widely used. Next slide. So you can see normal cervix on the left. They would have applied acetic acid, but it is a native squamous epithelium that is there, so it remains pink. And the cervix on the right side has precancerous changes. And once acetic acid is applied, you can see that it has coagulated the nuclear protein that is there, and you can see aceto-white areas, and those are the abnormal areas. Okay, next slide. So you can see positive aceto-white area starting from 12 o'clock to 12 o'clock position. Round the clock, around the cervical os, you can see that there is aceto-white area that is there. So this is the area that is abnormal, and the area outside that which remains pink is a normal area. So what we do is, in a higher method of screening, what is called as a colposcopy, which is a diagnostic testing, we put in a camera, magnify this image. Under acetic acid, we visualize the cervix under magnification, and we take biopsy from this aceto-white area and an iodine negative area. That gives us the diagnosis. Because if I have to go and take biopsy without applying acetic acid from this cervix, I would take from anywhere, even outside the area that is pink. But when I use acetic acid, it tells me exactly which area is abnormal, and I take a biopsy from this particular area, I am able to diagnose the problem. Next slide, please. So this is the iodine testing, villi. So you can see that the cervix on applying iodine has become completely mahogany brown, and that is the normal part. Yes. Next slide. But what is important is that for this via villi testing to be done, very good lighting is important. And via may be particularly be useful in developing countries where cytology is sometimes unreliable. You may not be having laboratories in the rural areas to report, or when you're doing large scale camps where practically reporting is not feasible and follow-up rates are very low and the resources are limited. Next slide, please. So the most common screening modality that has been practiced is the cervical cytology. So this is the standard and most successful screening method up till date, which has helped a lot in reducing the incidence of cervical cancer and reducing the mortality. Next slide. So what happens when someone undergoes a pap smear? So we put the patient in a dorsal position, make her comfortable, lie down with legs open. Your gynecologist will put in a speculum, open up the vagina and visualize the cervix. And then we use, yes, just like this. And we use a brush. Previously, we used to use a wooden spatula and just do a 360-degree rotation. It is kept at the level of cervical os, and a 360-degree rotation of the brush is done, and it is put in a liquid and sent for testing. That is called as liquid-based cytology. Previously, the same thing, instead of a brush, we used to use a wooden spatula and do a 360-degree rotation, make a smear on the slide and send it to pathologist for analysis. So what was happening when we did this method of creating smear was that many times we used to get reports like very thin smear, very thick smear, sometimes very less cervical cells. Sometimes, you know, the drying, when the drying or fixation of the smear is not done very properly, the reporting may be very difficult. So these kind of things and problems we were facing with the screening. And to do this, you need more manpower and you need more trained manpower. Next slide, please. So right now, in order to avoid this, we are using what is called as a liquid-based cytology, where we put in the brush and take a smear and put it in a liquid and send it for testing. So this is a very established method, and it has helped in reduction both incidence and mortality. in a lot of countries. And most of the countries which have a well organized screening programs are based on this particular method of screening. And in developing countries, limitation of the finances and manpower resources have inhibited this method of testing and national screening programs. Next slide. So what are all the problems associated with this method? You need skilled technical staff to take the smear. Exactly at this former columnar junction, it has to be taken. High cost. The pap smear now in a private laboratory costs about 800 rupees. And labor intensive testing and reporting of the smears is a problem. Inadequate follow-up of abnormal smears. So all these, many a time they give the smear, they go disappear for about 1 month and they don't come back at all. So people are very busy these days to follow up. So these are all the problems that we are facing. And in order to improve the results of the pap smears, we made the following improvements have been made. That is use of cytobrushes and liquid-based cytology and automated analysis. These are all the various methods that have been tried in order to improve the reporting. Next slide, please. So this is what I was talking about, liquid-based cytology. You can see the brush over there with the handle which is in blue and the tip that is in white. And we do a 360-degree rotation, put it in that bottle which has a liquid and send it for testing, cytology. Next slide. So by using LBC, liquid-based cytology, it is easier to read. We have avoided all this very inadequate smear and all those kind of reporting. Specimen quality has been improved and it has very high sensitivity in detection of problems when compared to conventional smears, and it's also cost effective. Next slide. And now everybody is only talking about HPV DNA testing. So, but the only problem with this is it has still not become cost effective and it is still not been implemented in many places. So why we are talking about HPV DNA testing now is that, pap smear, the reporting is subjective. One person that sees a smear on a microscope may feel it is normal, may feel it is low grade lesion, while in the eyes of another person, the same thing may look abnormal. The same thing may look like a high grade lesion. In order to avoid this, HPV DNA testing was brought up. Anyway, we know that HPV infection is the one that is causing these changes in the cervix. So instead of looking for the changes in the cervix, we just test for HPV there. This is just like your COVID-19 RT-PCR. Same concept is being done for HPV DNA testing. So why it is better when compared to the smears is that nobody needs to comment because it is subjective. There will be no discrepancy from person to person. If it is positive, it is positive. If it is negative, it is negative. So it is as simple as that. Next slide. So which is better, pap smear or HPV testing? Like I told you, the cytology results suffer from a degree of subjectivity while it does not happen with HPV testing. It's highly objective, it's positive for everybody, negative for everybody, and it's highly reproducible. How many ever times you run the testing on the same sample, it is the same. Is going to give the same report. And self-sampling is possible. What is self-sampling? Our women are very shy to come and get a pap smear testing done or a HPV testing done. We are distributing swabs in the community. Just like the COVID-19 swab, you teach them how to put it inside the vagina and take a swab. They will take the swab and send it to you. We can go to the community and collect it. This is the advantage of HPV testing. Self-sampling can be done. Women can test themselves. And if HPV testing is combined along with cytological screening, the screening interval can be safely increased. Both have been done and it is negative. Very safely, from screening every 3 years, you can safely increase it to even 8 to 10 years. That's what they're saying. Because HPV has a high negative predictive value. HPV comes out negative, you have no doubt, definite negative. Then you can screen the woman, provided the pap smear is also normal. Just come back after 10 years, I'll see you at that time. Next slide. So what is colposcopy and biopsy? It is a diagnostic modality. So once we do a via villi testing or a pap smear testing, it comes out to be a high grade lesion, and or somebody comes with a symptom, we put in a speculum, the cervix looks like it is abnormal. These are all the lesions when we conditions when we will go for a colposcopy. Colposcopy is nothing but a magnifying lens that is being used, a device that magnifies the cervix, it zooms the cervix and shows us, while we can examine it under acetic acid and lugols iodine and magnify the lesion, the astovite areas, and it will tell us various details about it, will tell us whether it's a high grade lesion or a low grade lesion, and very targeted biopsy of the area we can take from exactly the area that is showing changes of developing into a CIN 1, 2, 3 or a CA cervix. Targeted biopsy can be taken with the help of colposcopy, and sometimes even without a biopsy, a colposcopy tells us whether it is just a low grade lesion or a high grade lesion. So what are all the indications? Women with high grade squamous intraepithelial lesion on doing a pap smear, or women that has a low grade squamous intraepithelial lesion but it is not clearing and it is persisting more than 2 6-monthly assessments, and clinically looking suspicious cervix, multicentric disease. So these are all the conditions when we will do a colposcopy. So but the basic requirement is we need a good apparatus. Next slide please. So this is a binocular colposcope. What you are seeing, it is attached to a television. So the colposcope, the scopist will look into the cervix, will focus the cervix, magnify and look into the television for the changes. So here what you are seeing is a digital colposcope. This is what I have in my clinic. So a digital colposcope just has a lens and it is connected to a laptop. And as and when we keep zooming and applying acetic acid and lugols iodine, we can see the image of the cervix in very good detail. And we can take a targeted biopsy under the guidance of a colposcope. Next slide, please. So the take-home message today is that cervical cancer is a 100% preventable disease, and creating awareness about the screening and vaccination that is being available for cervical cancer is the only way forward in preventing this from being a public health problem. Next slide, please. So WHO has proposed to eliminate cervical cancer as a public health problem by 2030. By elimination, they mean an age-adjusted incidence risk of bringing it down to less than 4 in 1 lakh women by 2030. So how are we planning to achieve this? By 90% of girls should be fully vaccinated with this vaccine by the time they reach 15 years of age. At least 70% of the women should be screened with a high performance test between 35 to 45 years of age. Cancer should receive treatment and care. So this is how we plan to eliminate this as a public health problem. Next slide, please. So at Excella Gynae Care, we conducted a camp, a free camp for pap smear on occasion of Women's Day. But unfortunately, the attendance was very poor. So it is true that 1 woman dies of cervical cancer every minute, which means that by the time I have finished this presentation, at least 10 women have died of cervical cancer. And one small and painless test, that is pap smear test or a HPV test, can detect cervical cancer even before it starts. And all we need to tell women is that don't fear the smear. It is a simple diagnostic test and it can save lives. Next slide, please. So I will do anything to protect my girl child. Will you? Yes, I think every mother, everyone will do anything to protect their girl child. And what you should do if you have a girl child, if you have students or girls around you, educate them about HPV vaccination and do not wait at all. And this is how we are going to end cervical cancer, eliminate cervical cancer as a public health problem. Thank you very much for patient listening.
Thank you, ma'am, for the excellent session.
Yes.
It was very informative.
Thank you.
Thank you so much, ma'am, for the valuable information you have shared with us. If anybody having questions, please post your questions in the chat box.
Ma'am, there are questions in the chat
Ma'am,
box.
yes, there are some questions in the chat box. If both the partners are HPV negative, then what frequency of testing is appropriate? Every 8 to 10 years, if a person is HPV negative, 8 to 10 years, provided the cytology is also negative, we are just doing a HPV testing for every 5 years is good enough. And we can reduce the frequency of screening after a person reaches 60 years of age. Secondary infection of cancer, cervical cancer. Do you mean secondary prevention, surgical operation? Okay, I believe Mr. Rajkumar wants to know if for prevention of cervical cancer we can remove the uterus. Is that what you want to know, sir? No, we don't advise surgical removal of uterus or hysterectomy for preventing cancer of the uterus or cancers of the cervix. It is not done. Uh, when we have a preventive modality for cervical cancer, that is early detective modality, that like pap smear and HPV testing, what is the necessity to remove the uterus? Does that answer your question, sir? How HPV?
Yes, madam.
Yes. Okay, we don't have to do a hysterectomy in order to prevent cervical cancer. The preventive method would be to go for a regular screening test every 3 to 5 years, depending upon the age group. And once, if we happen to detect pre-cancerous lesion like CIN 1, 2, or 3 in women who are being screened, there are treatment methods what is called as loop excision. That is, under the colposcope we would see the areas that are abnormal and do a excision of the cervix, the part that is having the cervical cancerous changes. While in women who have completed their family, who have finished having children, they want to have 2 children, they have finished their family and they are old enough to undergo hysterectomy, if they are having CIN3, we can proceed with the hysterectomy. Yes, like you asked, in that case, we can proceed with the hysterectomy because CIN3 has a very high risk of converting into a cervical cancer very soon. But if it is early lesions like CIN1, many a times they regress. CIN1 and 2 have higher chances of regression back to normal. So in those cases, we don't have to do a surgical removal of uterus. Instead, we only keep them on regular follow-up and colposcopy. Thank you. Any more questions?
Dear participants, if you have any questions, you can post in the chat box or you can unmute yourself and ask.
Ma'am, how HPV testing is done in case of a man?
See, as of now, we are doing HPV testing only for women. But if at all it is in future, we are going to do screening for a man. It can be done from a swab from the genital region or urinary samples. But right now, we are not doing screening for men.
Okay, ma'am. Thank you.
Thank you.
If there are no questions from participants, can we conclude, ma'am? Aishari, ma'am?
Thank you, ma'am, for your informative session, and this was a very wonderful session, ma'am. Thank you very much, ma'am.
Thank you very much for giving me the opportunity to speak to all of you. Thank you.
Thank you, ma'am.
Shall we wind up these sessions, ma'am?
Yes, ma'am. You can give the announcement for tomorrow and then you can wind up.
Yes, ma'am. Thank you all for your patient hearing. Tomorrow the sessions will be started at 10:30. All are requested to log in before 5 minutes. And that's all for today. Please fill the feedback forms that is posted in the chat box. Thank you.
Thank you, ma'am. Thank you, participants.